There's a place on this earth so deep, so dark, and so utterly removed from the world we know, that for most of human history we simply assumed nothing could live there. We assumed the pressure would crush every living thing. We assumed the cold would still every biological process. We assumed the darkness would extinguish every spark of life before it ever had a chance to ignite. We assumed, in short, that this place was dead. That it was nothing more than an enormous, silent grave carved into the bottom of the ocean floor, a wound in the planet so severe and so ancient that even nature itself had given up on filling it with anything meaningful. We were wrong. We were profoundly, spectacularly, and almost embarrassingly wrong. What lies at the bottom of the Mariana Trench, at the very deepest point on the surface of this entire planet, is not emptiness. It is not silence in the way we once imagined silence. It is not a void. What lies down there, waiting in the crushing dark beneath nearly 11 kilometers of seawater, is life. Extraordinary, resilient, bewildering, almost unbelievable life. Life that has found ways to not merely survive, but to thrive in conditions that would destroy any organism we might consider familiar. Life that has rewritten the rule book on what biology is capable of. Life that has been quietly going about its business for millions of years while we, on the surface, looked down into the ocean and assumed there was nothing worth looking for. This is the story of the Mariana Trench. This is the story of the Challenger Deep, the single deepest point ever measured on the surface of this earth, a location so extreme it sits nearly 7 miles beneath the surface of the Pacific Ocean. This is the story of the machines built to reach it, the scientists brave enough to descend toward it, and the living world they discovered waiting for them when they arrived. This is the story of what happens when human curiosity refuses to accept a boundary, even one drawn at the very bottom of the world. And this is, perhaps most importantly, the story of how much we still do not know. Because for all the progress that has been made, for all the remarkable descents and the extraordinary footage and the astonishing biological specimens hauled up from those crushing depths, the truth is that the Mariana Trench remains, in almost every meaningful sense, unexplored. The truth is that we have seen only the narrowest sliver of what lies down there. The truth is that every descent raises more questions than it answers, and that the trench, vast and patient and ancient, is keeping the vast majority of its secrets still locked away in the dark. So let us begin. Let us start at the surface of the Pacific Ocean, on a day when the water looks almost ordinary, when the waves are manageable and the sky is open and there is nothing visible to suggest that directly beneath this unremarkable stretch of open sea, the floor of the planet drops away into the deepest hole in the world. Let us start with what the trench is, how it came to be, and why for so long we thought we understood it, and then let us go deeper. To understand the Mariana Trench, you first have to understand the planet you are standing on, because the trench is not an accident. It is not a random feature. It is the direct result of forces so enormous and so ancient that our human minds struggle to truly comprehend their scale. The trench is a scar left by the collision of tectonic plates, those massive, slow-moving sections of the Earth's crust that drift across the surface of the planet over millions of years, carrying continents and ocean floors with them like passengers on the slowest boats imaginable. The Pacific Ocean sits on what geologists call the Pacific Plate, one of the largest tectonic plates on Earth. To the west of this plate, pressing against it with a force that has been building for tens of millions of years, sits the Mariana Plate. When these two plates meet, something dramatic happens. The Pacific Plate, which is older and denser and slightly heavier, does not simply stop. It does not pile up against the Mariana Plate the way two cars might crumple against each other in a collision. Instead, it does something far more dramatic. It dives. It slides beneath the Mariana Plate in a process that geologists call subduction, and as it does, it drags the ocean floor down with it, pulling the crust of the Earth downward at an angle that creates a long, narrow, extraordinarily deep trench at the point where the diving begins. This process has been underway for a very long time. The subduction zone that created the Mariana Trench has been active for somewhere in the neighborhood of 180 million years. 180 million years of the Pacific Plate grinding steadily downward beneath the Mariana Plate. 180 million years of the ocean floor being dragged deeper and deeper into the Earth's mantle. 180 million years of slow, grinding geological violence producing the deepest feature on the surface of the planet. The result is a trench that stretches for roughly 2,500 kilometers, that is approximately 1,500 miles, in a crescent shape through the western Pacific Ocean, located about 200 kilometers east of the Mariana Islands. It is, in terms of sheer scale, a staggering feature. If you were to take the tallest mountain on Earth, Mount Everest, standing at 8,849 meters above sea level, and place it at the bottom of the Challenger Deep, the deepest point in the Mariana Trench, the summit of that mountain would still be more than a mile beneath the surface of the ocean. The trench is not just deep. It is almost incomprehensibly deep, deep in a way that challenges the human imagination to truly picture. The Challenger Deep itself, that specific point within the Mariana Trench that represents the lowest elevation on the surface of this planet, sits at approximately 10,935 meters below sea level. That measurement has been refined and refined over decades as surveying technology has improved, and there is still some debate among scientists about the precise number. But regardless of the exact figure, the essential truth is the same. This is the deepest place on Earth. This is the floor of the world, and for most of human history, it was as unknown to us as the surface of the moon. The first time anyone knew this trench existed at all was in 1875, when a British naval vessel called HMS Challenger was conducting one of the earliest systematic surveys of the world's oceans. The Challenger was equipped with sounding lines, essentially very long ropes weighted at the end, that could be lowered into the water to measure depth. When the crew lowered their sounding line over a particular patch of the western Pacific, the line kept going down and down and down, paying out farther than it had at any previous location in their survey. When they finally hit bottom and hauled the line back up, they had recorded a depth of approximately 8,184 meters. This was, at the time, the deepest point ever measured. The trench was named after the ship that found it, and the deepest point within the trench was named Challenger Deep in honor of that original discovery. But of course, a sounding line, however impressive for its era, tells you almost nothing beyond depth. It tells you the bottom is down there somewhere. It does not tell you what the bottom looks like, what lives on it, what the conditions are, what the temperature is, what the pressure feels like, what the sediment is made of, or any of the 1,000 other questions that a curious mind might ask. For those answers, you would need to go there yourself. And going there yourself presented a set of challenges so formidable that it would take nearly another century before anyone was able to attempt it. The fundamental challenge of reaching the Challenger Deep is not distance. In horizontal terms, the location is not particularly remote. It sits in a relatively accessible part of the Pacific. The challenge is vertical. The challenge is that to get to the bottom of the Challenger Deep, you have to descend through nearly 11 kilometers of water, and as you descend, the pressure of all that water above you increases steadily and remorselessly. At sea level, the pressure of the atmosphere on your body is approximately 1 atmosphere, a unit of measurement we use precisely because it represents the normal atmospheric pressure at Earth's surface. As you descend into the ocean, pressure increases by roughly 1 additional atmosphere for every 10 meters of depth. Do the math for the Challenger Deep, and you arrive at a pressure of approximately 1,096 atmospheres at the bottom. 1,096 times the pressure you feel standing on the surface of the Earth. That is a pressure of roughly 16,000 pounds per square inch, more than enough to collapse any structure not specifically engineered to withstand it, and more than enough to kill any unprotected human being almost instantaneously. Building a vessel capable of withstanding that pressure while also being able to move through the water, carry instruments, collect samples, and safely return to the surface is an engineering challenge of the first order. The first people to meet that challenge were the Swiss physicist Auguste Piccard and his son Jacques, who designed and built a vessel called the bathyscaphe Trieste. The Trieste was an ungainly-looking machine. It resembled, in some ways, a large buoy with a small sphere attached to its underside. The sphere was the pressure vessel, a thick-walled steel ball where the crew would ride. The large float above it was filled with gasoline, which is lighter than water and provided buoyancy. Iron pellets provided ballast to take the craft down. To ascend, the iron pellets could be released, allowing the gasoline-filled float to carry the sphere back toward the surface. It was in this machine, on the 23rd of January, 1960, that Jacques Piccard and United States Navy Lieutenant Don Walsh made the most extraordinary dive in human history to that point. They descended through nearly 11 kilometers of water, through cold and darkness and increasing pressure, and they reached the bottom of the Challenger deep. They sat there for about 20 minutes. They could see almost nothing through the small viewport. The descent had stirred up too much sediment. But they reported seeing what appeared to be flatfish lighters on the seafloor, which caused immediate and significant scientific controversy, because many scientists at the time believed that nothing at all could live at such depths. The pressure, the argument went, would prevent the biochemical processes necessary for life from functioning. Life, according to this view, simply could not exist in such a place. Walsh and Piccard's observation of apparent life at the bottom of the Challenger deep was the first crack in that assumption. It would not be the last. Over the following decades, as technology improved and as more missions were mounted to deep ocean environments, not necessarily to the Challenger deep itself, but to various deep-sea locations around the world, the assumption that the deep ocean was barren and lifeless began to erode. First slowly, then with increasing speed, as evidence accumulated that the deep sea was not an empty void but a complex, diverse, and extraordinarily productive ecosystem unlike anything we had encountered before. But reaching the Challenger deep specifically, that deepest point, that ultimate extreme, remained extraordinarily difficult. The Trieste dive of 1960 was followed by a very long gap. The logistical and engineering challenges of building a vessel capable of surviving that pressure were simply too great for most programs. There were deep ocean research submersibles built and operated during the following decades, but most of them were designed to operate at much shallower depths, a few hundred meters, perhaps a few thousand. Full ocean depth capability, meaning the ability to reach the absolute bottom of the Challenger deep, remained beyond the reach of most programs for decades. Not until 1995 that the next significant unmanned visit to the Challenger deep was made by a Japanese remotely operated vehicle called Kaiko. And it was not until 2012 that another crewed vehicle reached the bottom, when filmmaker and explorer James Cameron descended in a specially designed submersible called the Deep-Sea Challenger. Cameron's dive was significant not only because it was the first solo crewed descent to the Challenger deep, but also because he brought with him an array of scientific instruments and cameras capable of documenting what he found in unprecedented detail. But even Cameron's extraordinary dive, for all its achievements, was limited by the technology of its time. The Deep-Sea Challenger was a remarkable machine, but it had limitations in terms of how long it could stay at depth, how far it could travel across the seafloor, and how much it could carry or collect. The picture it painted of the Challenger deep, while more detailed than anything that had come before, was still the picture of a single point visited for a limited time. The mission that truly began to change our understanding of the Mariana Trench, the mission that provided the foundation for much of what we know today, the mission whose findings form the core of this documentary, is the series of dives conducted by the Fendouzhe. And to understand why the Fendouzhe matters, you first need to understand what it is and what it was designed to do. The Fendouzhe, whose name translates roughly as striver in English, is a full ocean depth crewed submersible. It is capable of carrying 3 people to the very deepest points in the world's oceans, and it has done so repeatedly. Unlike many of its predecessors, the Fendouzhe was designed not just for a single historic descent, but for sustained scientific operations at extreme depth. It was built to go to the bottom of the ocean not once, not twice, but again and again, each time carrying scientists and instruments capable of making careful, systematic observations of the environment at the bottom of the world. The Fendouzhe is a product of years of engineering development. Building a vessel that can withstand the pressure at the bottom of the Challenger deep requires materials and manufacturing techniques of extraordinary precision. The pressure hull, the sphere inside which the crew lives and works during a dive, must be perfectly round, because any deviation from perfect sphericity creates stress concentrations that can lead to catastrophic failure under pressure. The materials must be strong enough to withstand forces that would crush conventional steel like paper. The viewports must be made of a special transparent material thick enough to resist the pressure while still allowing the crew to see what is outside. Every penetration of the hull, every cable, every wire, every pipe passing through the wall of the pressure vessel must be sealed with absolute perfection, because a single failed seal at those depths would allow water to enter with the force of a small explosion. Beyond the structural challenges, the Fendouzhe had to be equipped with a complete suite of scientific instruments capable of operating at extreme depth. Water samplers, sediment corers, biological collection equipment, high-definition cameras, sonar systems, chemical analyzers, all of these instruments had to be designed or adapted to function reliably under pressure conditions that would destroy most standard equipment. The engineering effort required to produce a machine like the Fendouzhe is enormous, and it represents the culmination of decades of learning about how to build things that can survive at the bottom of the ocean. When the Fendouzhe began its series of dives into the Mariana Trench, it carried with it not just human crew members, but a new generation of scientific capability. Its cameras could capture high-definition video in the essentially complete darkness of the deep sea, using powerful lights to illuminate a world that had never been illuminated before. Its instruments could measure the chemistry of the water at depth, sampling the water column as the submersible descended and analyzing what they found for evidence of chemical processes taking place in the deep ocean. Its collection equipment could gather sediment samples, water samples, and biological specimens for later analysis in surface laboratories. And its sonar could map the seafloor in ways that previous missions had never managed, building up a picture of the terrain at the bottom of the trench that was far more detailed than anything previously available. What the Fendouzhe found when it reached the bottom of the Mariana Trench, what it documented over repeated dives spanning multiple years, is a story so remarkable, so filled with unexpected discoveries and paradigm-shifting revelations, that it is difficult to know exactly where to begin. Perhaps the best place to start is also the most fundamental: life. Life at the bottom of the Mariana Trench should not exist. That at least was the conventional wisdom for a very long time, and it was not an unreasonable position. The conditions at the bottom of the Challenger deep are, by almost any measure, among the most extreme that any living organism on Earth is ever required to face. The pressure, as we have already established, is roughly 1,100 times the pressure at the surface. The temperature hovers just above the freezing point of water, typically somewhere around 2 to 4 degrees Celsius. There is no sunlight, none at all, not even the faintest trace. The last photon of sunlight is absorbed within the upper few hundred meters of the water column, and below that, as you descend through the thousands of meters to the bottom of the trench, there is nothing, complete, absolute, and eternal darkness. And yet when the lights of the Fendouzhe illuminated the seafloor at the bottom of the Challenger deep, what they revealed was not emptiness. What they revealed, in the narrow cone of light that the submersible's powerful LEDs projected against the sediment, was life. Not the sparse, isolated, barely there life that some scientists had begun to suspect might cling to existence in even the most extreme environments. Not just a few hardy microbes making a meager living in the sediment, but actual, visible, abundant, diverse life. A whole ecosystem hidden in the dark, going about its business in complete ignorance of the fact that it was supposedly impossible. The seafloor at the Challenger Deep is covered in soft sediment, fine particles that have drifted down from the water column above over millions of years, accumulating in layers to form a thick, soft substrate that feels, from the footage captured by submersibles, almost like fresh snow. This sediment is not sterile. Far from it, it is alive with microorganisms, bacteria, archaea, and other microscopic life forms, in concentrations that surprised researchers who expected the deep, nutrient-poor environment to support only sparse microbial communities. But the microbial life, remarkable as it is, was almost overshadowed by the visible fauna. The creatures that can actually be seen in the submersible footage, the animals large enough to register on a camera, the organisms that could be watched going about their lives in the deep dark. Among the most striking of these were the amphipods. Amphipods are crustaceans, small, shrimp-like animals that are found throughout the world's oceans, from coastal tide pools to the open sea. In most environments, they are small, typically a centimeter or two in length, and somewhat unremarkable in appearance. But the amphipods of the Mariana Trench are something entirely different. In the deep, cold, pressurized waters of the trench, these animals grow to sizes that seem almost grotesque compared to their shallow water relatives. Individuals measuring 15, 20, even 30 centimeters in length, nearly a foot, have been documented in the Challenger Deep and throughout the Mariana Trench. These giant amphipods, which belong to a genus called Hyaerndalea among others, are extraordinarily abundant in the deep trench environment. They can be seen in submersible footage swarming over the seafloor in numbers that are almost hard to process, hundreds of individuals visible in a single camera frame, crawling and swimming and feeding with the frantic energy of animals that know that food in this environment is not something to be wasted. What are these amphipods eating? This is a crucial question because it gets at the heart of how any ecosystem can function at the bottom of the Mariana Trench. In the sunlit surface waters of the ocean, the foundation of the food web is photosynthesis. Tiny marine plants and algae use the energy of sunlight to convert carbon dioxide and water into organic matter, which then passes up the food chain as it is eaten and digested and transformed. But at the bottom of the Mariana Trench, there is no sunlight. There can be no photosynthesis. The energy input that drives the ecosystem at the surface simply does not exist down here. So where does the energy come from? Part of the answer is what oceanographers call marine snow. This is a term for the constant, gentle rain of organic particles that falls from the surface ocean downward through the water column. Dead phytoplankton, dead zooplankton, fragments of fecal material, bits of organic debris, all of this drifts downward slowly, through thousands of meters of water, and some portion of it eventually reaches the seafloor. In shallower environments, most of this organic material is consumed by animals higher in the water column before it can reach the bottom. But in the Mariana Trench, the enormous depth means that material falling from the surface has a very long way to travel before it reaches the floor. By the time it gets there, much of it has been consumed or degraded. The deep sea floor receives a very thin, very dilute supply of organic material from above, and yet those amphipods are thriving. Those communities of animals are dense and diverse. Something is providing enough energy to support them. Part of the answer lies in the funneling effect of the trench's geometry. The Mariana Trench is long and narrow, with steep walls on either side. Organic material that settles out of the water column over the broad area of the trench does not simply land wherever it falls. It tends to be channeled by currents and by the V-shaped geometry of the trench itself toward the deepest point at the bottom. Think of it as a funnel. Material falling across a wide area converges as it descends and concentrates at the deepest point. This concentration effect means that the sediment at the bottom of the trench is actually richer in organic material than you might expect given how far it is from the surface. The very geometry of the trench, the feature that makes it so extreme, also makes it, in a sense, a collector of the nutritional material that drifts down from above. But organic material settling from above is not the only energy source driving the deep sea ecosystem. Far from it. The other major energy source, and in some ways the more extraordinary one, is chemosynthesis. Chemosynthesis is the process by which certain microorganisms can generate energy not from sunlight, but from chemical reactions. Specifically, these organisms can harvest the energy released when inorganic compounds are oxidized, when hydrogen sulfide is converted to sulfur, for example, or when methane is oxidized, or when various other chemical reactions occur that release energy. These chemosynthetic microorganisms form the base of a completely separate food chain from the photosynthetic one we are more familiar with. They are, in a sense, the primary producers of the deep sea, the organisms that take raw chemical energy and convert it into the biological material that other organisms can then consume. Chemosynthesis was not discovered until 1977, when oceanographers exploring the deep ocean near the Galapagos Islands encountered something that absolutely nobody expected: hydrothermal vents. These are places on the ocean floor where seawater seeps down into cracks in the crust, is heated by the underlying magma, absorbs minerals and chemicals from the surrounding rock, and then vents back out into the ocean as superheated, chemically rich fluid. Around these vents, researchers were astonished to find entire ecosystems, tube worms, crabs, clams, fish, shrimp, living in complete independence from the sun. The energy driving these ecosystems came not from photosynthesis but from the chemical energy in the vent fluids, captured by chemosynthetic bacteria that lived in the vent environment and formed the base of the food web. The discovery of hydrothermal vent ecosystems was one of the most significant biological discoveries of the 20th century. It demonstrated beyond any reasonable doubt that life could exist and even flourish in the complete absence of sunlight, given a suitable chemical energy source. It fundamentally changed our understanding of the limits of life and the range of environments in which biology can operate. The Mariana Trench does not appear to have major hydrothermal vents at its deepest points. The geology of subduction zones is different from the geology of mid-ocean ridges where vents are most commonly found. But the trench does have something related: areas where cold seeps occur, where fluids enriched in methane and hydrogen sulfide slowly percolate up through the sediment. These cold seeps are far less dramatic than hydrothermal vents. The fluids are not superheated. They do not form the dramatic black smoker chimneys that make hydrothermal vents so visually striking, but they provide a similar chemical energy source that supports chemosynthetic microbial communities. And in the sediment throughout the deep trench environment, chemosynthetic microorganisms are present and active, converting chemical energy into biological material that forms part of the foundation of the deep sea food web. What the Fendouzhe documented at the bottom of the Mariana Trench was thus not a single, simple ecosystem, but a complex, layered one. There were the microbial communities in the sediment, both heterotrophic organisms consuming the organic material that drifted down from above, and chemosynthetic organisms exploiting chemical energy sources in the sediment and water. There were the amphipods and other larger invertebrates that fed on this microbial base and on the organic material in the sediment. There were fish, yes, fish, actual fish with fins and scales and eyes, living at these extraordinary depths, feeding on the invertebrate communities below them. And there were other creatures, strange and unexpected and deeply fascinating, that did not fit neatly into any of the familiar categories. Among the fish documented in the Mariana Trench, perhaps the most remarkable are the Hadal snailfish, creatures belonging to the family Leporidae that have colonized the deepest trenches of the world's oceans and become, in the process, some of the most extreme organisms on Earth. Snailfish look, at first glance, like small, pale, almost translucent fish. They have a distinctive tadpole-like body shape, with a large head tapering into a long, thin tail. They are not particularly large. Most individuals are between 15 and 30 centimeters in length, but their appearance is deceptive because these modest-looking animals are living at depths that no other fish species can tolerate. The The snailfish of the Hadal zone, the zone below 6,000 meters that encompasses the world's deepest trenches, have undergone a remarkable suite of adaptations that allow them to survive at pressures that would kill any other vertebrate. These adaptations are not superficial. They reach down to the molecular level, to the very biochemistry of the cells that make up the fish's body. And understanding these adaptations requires understanding something about what pressure actually does to living tissue. When you subject a living cell to very high pressure, a number of things happen, and most of them are bad from the cell's point of view. Proteins, the molecules that carry out most of the chemical work inside cells, catalyzing reactions, maintaining structure, transmitting signals, are extremely sensitive to pressure. Under high pressure, the three-dimensional shapes of protein molecules are distorted. Enzymes lose their ability to catalyze the chemical reactions they are supposed to facilitate. Cell membranes, which are made of lipid molecules arranged in a specific way, become too rigid under pressure to function properly. The physical properties of water itself change under high pressure, affecting how molecules dissolve and interact with each other. In short, the biochemistry of life, the intricate, precisely tuned machinery of cellular processes, does not work properly under the pressures found at the bottom of the Mariana Trench. Or rather, it does not work properly without modification. The trick that deep-sea organisms have evolved is to modify that biochemistry in ways that compensate for the effects of pressure. One of the most important of these modifications involves a molecule called trimethylamine oxide, or TMAO. TMAO is a small organic compound that is found in the tissues of many marine animals, particularly fish, and it is known to act as a piezolyte, a molecule that protects proteins from the damaging effects of pressure. What TMAO appears to do is stabilize protein structures under high pressure, preventing the pressure-induced distortions that would otherwise render proteins non-functional. In shallow water fish, TMAO is present in relatively low concentrations, but in the snailfish and other deep-sea fish, the concentration of TMAO in the tissues increases with depth. The deeper the fish lives, the more TMAO it contains. At the depths found in the Mariana Trench, the TMAO concentration in snailfish tissues is so high that the fish reportedly have a distinctly strong smell when brought to the surface. TMAO breaks down into trimethylamine, a compound responsible for the characteristic odor of fish. But TMAO is not the only molecular adaptation. Deep-sea organisms also modify the lipid composition of their cell membranes to maintain the right degree of fluidity under high pressure. In shallow water organisms, cell membranes contain a specific ratio of saturated and unsaturated fatty acids that keeps the membrane at the right consistency at normal pressures. Under high pressure, membranes would normally become too rigid to function. Deep-sea organisms counteract this by incorporating more unsaturated fatty acids into their membranes, which remain more fluid under pressure. This adaptation, which is the deep-sea equivalent of what animals living in cold environments do to prevent their membranes from becoming too rigid in the cold, allows deep-sea organisms to maintain functional membranes even under the crushing pressures of the Mariana Trench. These molecular adaptations are fascinating in their own right, but they point to something even more fundamental and philosophically profound. They tell us that life does not simply resist the environment in which it finds itself. It does not brute force its way through the challenge of extreme conditions. Instead, it adapts. It changes itself at the most basic level of its chemistry to align with the environment rather than fight against it. The snailfish does not resist the pressure at the bottom of the Mariana Trench. It has become a pressure-adapted organism. The crushing force that would kill almost any other animal has been incorporated, over millions of years of evolution, into the fundamental design of the fish's body. The fish and the pressure are not adversaries. They have reached, through the long process of evolution, a kind of accommodation. This insight that life adapts to align with extreme conditions rather than simply enduring them is one of the most important themes that emerges from the study of the Mariana Trench ecosystem. It appears again and again as you look at different organisms and different challenges. The amphipods that swarm the deep trench floor have not simply refused to be crushed. Their bodies have been redesigned at the molecular level to function under pressure. The bacteria in the sediment have not simply managed to survive in the cold. Their enzymes have been shaped by evolution to work efficiently at temperatures that would freeze the enzymes of surface-dwelling bacteria into uselessness. Life, it turns out, is not a fixed thing that encounters the environment and either survives or dies. Life is a process, a dynamic, ongoing negotiation between organism and environment that has been running for billions of years and that has produced, through the mechanism of natural selection, an extraordinary range of solutions to an extraordinary range of environmental challenges. The Mariana Trench is simply the most extreme version of this story that we have found on our own planet so far. It is the case study that pushes the story of adaptation and survival to its absolute limit. Understanding who lives in the Mariana Trench and how they survive is one thing. Understanding how they are organized, how they interact with each other, how energy and nutrients flow through the ecosystem is another, and it is a question that the repeated dives of the Fendouzhe and other deep-sea missions have begun to address in remarkable ways. The deep-sea ecosystem of the Mariana Trench is, in many ways, fundamentally different from the ecosystems we are most familiar with at the surface. Surface ecosystems, whether they are forests, grasslands, coral reefs, or open ocean, are driven by the constant input of solar energy through photosynthesis. This creates a rich, abundant food supply that supports a dense and diverse community of organisms arranged in a complex food web. The deep-sea ecosystem, by contrast, is driven by a much more limited and irregular energy supply. The organic material that drifts down from above is sparse and unpredictable. The chemical energy available from chemosynthesis, while important, is also limited. The deep-sea ecosystem is, in energy terms, a very lean place to live. This energy limitation has profound consequences for the organisms that live there and for the structure of the ecosystem as a whole. In an energy-rich surface environment, organisms can afford to be relatively extravagant in their biology. They can grow quickly, reproduce frequently, maintain complex bodies with high metabolic rates, and generally live at a fast pace. In the energy-poor deep sea, none of this is possible. Deep-sea organisms tend to grow very slowly. Some deep-sea fish grow so slowly that individuals can live for decades, even a century or more, reaching old ages that would be extraordinary in a surface-dwelling species of comparable size. They reproduce infrequently and invest heavily in each individual offspring. Their metabolisms are often dramatically reduced compared to surface-dwelling relatives, ticking over at the slowest pace compatible with maintaining life. This slow pace of life gives the deep-sea ecosystem a very different character from the bustling, energy-rich communities of the surface. It is a community adapted to waiting, to making the most of whatever comes along, to holding on through the long periods between nutritional windfalls with a minimum expenditure of energy. And then when something good does come along, when a significant parcel of organic material arrives from above, when a chemical opportunity presents itself, the response can be remarkably swift and vigorous. The most dramatic example of this dynamic, and one of the most extraordinary phenomena documented in deep-sea research, is what happens when a whale dies. Whales are the largest animals on Earth. An adult blue whale can weigh as much as 200 tons. When a whale dies at sea, its enormous carcass eventually sinks to the ocean floor. In shallow coastal waters, this event feeds a predictable and well-studied succession of scavengers, sharks, hagfish, crabs, and others that strip the flesh from the bones relatively quickly. But when a whale sinks into the deep sea, and given that whales spend much of their time in waters of significant depth, this happens regularly. What follows is an extraordinary cascade of biological activity that can persist for decades. The arrival of a whale carcass on the deep sea floor is, in the economy of the deep sea, the equivalent of a lottery jackpot. A single adult blue whale carcass delivers an enormous package of organic material, flesh, blubber, bone, and all the associated organic compounds stored in these tissues, to an environment that is typically very organic material poor. The energy content of a large whale carcass is roughly equivalent to the amount of organic material that would otherwise drift down from the surface and reach the deep sea floor over the span of 2,000 years. 2,000 years worth of marine snow, all arriving at once in one concentrated location. The biological response to this event unfolds in phases, each one giving way to the next as different groups of organisms exploit different aspects of the carcass. The first phase, the mobile scavenger phase, begins within hours to days of the carcass reaching the seafloor. Organisms capable of detecting the chemical signals released by the decaying flesh, the amphipods, the hagfish, the deep-sea fish, converge on the carcass from their surrounding environment, drawn by the trace of organic compounds drifting through the water. The speed of this convergence is remarkable given how sparse these organisms are in the surrounding environment. It suggests that deep-sea scavengers maintain a constant low-level search for exactly this kind of chemical signal, covering large distances in their search for food and responding quickly when they detect it. Within a relatively short period, large numbers of these organisms can strip the soft tissue from the whale's bones, consuming the flesh and blubber with a voracious efficiency that reflects the rarity of such opportunities. The second phase, the enrichment opportunist phase, follows as the soft tissue is depleted. During this phase, the exposed bones and the sediment immediately surrounding the carcass are colonized by organisms that exploit the organic material remaining in the sediment and in the bones themselves. Polychaete worms, crustaceans, and various other invertebrates are characteristic of this phase. They feed on the bacterial mats growing on the remaining organic material and process the enriched sediment for its nutritional content. But it is the third phase that is perhaps the most extraordinary, the sulfophilic phase, in which the whale bones themselves become the substrate for an entirely different kind of ecosystem. Whale bones contain large amounts of lipids, fats, stored deep within the bone matrix. As the bones slowly decompose, these lipids are metabolized by bacteria, and some of the metabolic products include hydrogen sulfide. This creates, at the site of the whale carcass, a local chemosynthetic environment, a zone where hydrogen sulfide is being produced and where chemosynthetic bacteria can thrive using it as an energy source. Remarkably, the organisms that colonize whale bones in this phase include many of the same species found at hydrothermal vents and cold seeps, organisms that are otherwise adapted to precisely the kind of chemosynthetic environment that the decomposing whale creates. A whale carcass on the deep sea floor becomes, in effect, a temporary, small-scale version of a hydrothermal vent ecosystem, supporting a community of chemosynthetically based organisms that may persist for decades as the bones slowly decompose. The whale fall ecosystem, as these carcass-based communities have come to be called, is one of the most profound demonstrations of the interconnectedness of the ocean. It shows how the deep-sea ecosystem, apparently isolated in its dark and pressurized world, is actually intimately connected to the surface ocean above it. The whales that swim through the sunlit surface waters, feeding on the abundant life there, are themselves serving as biological conveyor belts, packaging the photosynthetically derived energy of the surface ocean into their enormous bodies, and then, when they die, delivering that energy as a concentrated package to the deep sea floor. The surface and the deep are not separate worlds. They are part of the same system, connected by the slow rain of organic matter from above and punctuated by the dramatic deliveries of whale falls and other large carcasses. The Fendouzhe documented the aftermath of whale fall events during its dives in the Mariana Trench and surrounding deep-sea environments, and the footage is extraordinary. The bones of a whale lying on the sediment at extreme depth, covered in the whitish mat of chemosynthetic bacteria, surrounded by the specialized organisms that have colonized this temporary oasis. It looks, in the submersible's lights, like something between a cemetery and a garden. It is beautiful in its way and deeply moving because it embodies something fundamental about how life works. That nothing is wasted, that death feeds life, that even the most extreme and apparently inhospitable environments can be transformed into vibrant, productive communities given the right input of energy and nutrients. Among the most striking findings to emerge from the deep-sea exploration conducted in recent years, and one that sits uncomfortably alongside the wonder and beauty of the deep-sea ecosystem, is the discovery of microplastics in the sediment at the bottom of the Mariana Trench. Microplastics are tiny particles of plastic, typically defined as particles smaller than 5 millimeters, though many are far smaller, in the micrometer or even nanometer size range, that are found throughout the world's oceans as a result of the breakdown of larger plastic items and the direct release of small plastic particles from various products and industrial processes. We have known for some years that microplastics are widespread in the surface ocean, carried by currents and distributed across vast areas. We have known that marine animals ingest them, that they enter food webs, and that they accumulate in animal tissues. But the discovery of microplastics in the sediment at the very bottom of the Mariana Trench, at the deepest point on Earth, in one of the most remote and seemingly pristine environments on the planet, was a finding that sent a very clear and very uncomfortable message about the reach of human activity. The microplastics in the deep-sea sediment arrived there by the same processes that deliver organic material to the deep-sea floor. Small plastic particles in the surface ocean are colonized by microorganisms, which make them denser and cause them to sink. They aggregate with other particles, forming larger clumps that sink more rapidly. They are ingested by animals in the water column and packaged into fecal pellets that sink to the bottom. Through a combination of these processes, plastic material that enters the ocean at the surface eventually finds its way to the seafloor, and because of the funneling effect of the trench's geometry that we discussed earlier, the concentration of microplastics at the bottom of the Mariana Trench is actually higher than in the surrounding deep-sea environment. Scientists who analyzed sediment samples collected from the Challenger deep found microplastic particles throughout the sediment column. They found fibers, small threads of plastic material that likely came from synthetic clothing washed in household laundries, releasing tiny fibers with every wash cycle. They found fragments, small pieces of harder plastic that likely originated from larger items that had broken down over time. They found particles made of a wide variety of plastic types, reflecting the diversity of plastic materials we produce and use. The implications of this finding are significant on several levels. At the most basic level, it tells us that the deep ocean is not isolated from the effects of what we do at the surface. There is no remote enough place, no deep enough trench, no sufficiently extreme environment that is beyond the reach of our influence. The particles we release through our waste, through our industrial processes, through the slow degradation of the plastic objects that surround us in our daily lives, travel through the ocean and accumulate even in the deepest and most inaccessible corners of the planet. What these microplastics are doing to the organisms living at the bottom of the Mariana Trench is a question that researchers are still working to understand. We know that microplastics can cause physical harm when ingested. They can block digestive systems, damage tissues, and take up space in the gut that should be occupied by food. We know that plastics can carry toxic chemicals that are absorbed from the surrounding water and concentrated on the plastic surface, and that these chemicals can leach into the tissues of organisms that ingest the plastic. And we know that microplastics can affect the behavior of organisms, interfering with feeding and reproduction and other vital functions. Whether and how these effects manifest in the extreme, slow-paced, energy-limited ecosystem of the deep Mariana Trench is something that current science is only beginning to address. The organisms living at the bottom of the trench are already operating at the edge of what is biologically possible. They have evolved to squeeze every available nutrient out of a sparse environment and to maintain their biochemistry under crushing pressure with carefully tuned molecular adaptations. The addition of a novel contaminant, however small the individual particles might be, introduces an unknown into a system already pushed to its limits. What is clear is that the finding of microplastics at the bottom of the Mariana Trench is not just a data point about ocean pollution. It is a statement about the nature of our relationship with the planet. It is evidence that there is no longer anywhere on Earth that can be considered truly untouched by human activity. And in a place like the Mariana Trench, a place of such extraordinary biological richness, such astonishing adaptation, such irreplaceable scientific value, that fact carries a particular weight. There is something that happens to a person, apparently, when they are descending toward the bottom of the Mariana Trench in a submersible. As the craft sinks through the water, as the sunlight fades and then disappears entirely, as the world above becomes an increasingly distant memory and the darkness outside the viewports becomes absolute, there is a shift in perspective that is difficult to fully articulate, but that every person who has made this journey seems to remark upon. Part of it is the sheer physical sensation of isolation, being enclosed in a small sphere of air and light, suspended in an enormous volume of dark water, with the surface miles above and the bottom still miles below. Part of it is the intellectual weight of the knowledge that you are descending to the most extreme environment on Earth, that the pressure outside the hull is increasing with every passing minute, that the physical margin between survival and catastrophe is the thickness of the pressure vessel walls. Part of it is the strange, dislocating beauty of the bioluminescence, the living light of the ocean's creatures, which flares and drifts and pulses in the darkness outside the viewport as the submersible descends, providing the only illumination in a world without sun. And part of it, perhaps the largest part, is the growing awareness of scale, the awareness that you are descending through something enormous. That the ocean is not a surface feature, not just a skin of water stretched across the planet, but a volume. A vast, three-dimensional volume of water that constitutes the majority of the habitable space on Earth. The habitable volume of the oceans, the space within the ocean where organisms can live, is roughly 300 times larger than all the terrestrial habitats on Earth combined. Deep sea, the part of the ocean below 200 meters, accounts for the vast majority of this volume. And the Hadal zone, the depths below 6,000 meters that encompass the deep trenches, is the most extreme and least explored portion of this already vast and poorly understood space. This sense of scale, of descending through something almost incomprehensibly large into something almost incomprehensibly deep, is part of what makes the Mariana Trench so philosophically significant. It forces a reckoning with the limitations of human perspective, with the narrowness of the world we normally inhabit and understand. We live our lives in a thin layer of atmosphere and surface, separated from the vast majority of the habitable space on our own planet by a barrier, the ocean's surface, that we can cross easily in either direction, but rarely choose to descend through for more than a few tens of meters. The deep ocean is our planet's largest living space, and it is almost entirely unknown to us. This is not for lack of interest, it is for lack of capability. The engineering challenge of reaching the deep ocean is genuinely formidable, and even with modern technology, the ability to explore the deep ocean systematically is severely limited. A single dive of the Fendouzhe to the Challenger deep takes the better part of a day. The descent alone, at the pace that safety requires, takes several hours. Time at the bottom is limited by the life support capacity of the submersible, the amount of oxygen and other consumables it can carry. The ascent takes another several hours, and during all that time, the submersible can cover only a limited area of the seafloor, a few kilometers at most, given the need to move slowly and carefully in an environment where visibility is limited to the range of the vehicle's lights. Consider what this means for the task of mapping and exploring the Mariana Trench. The trench stretches for approximately 2,500 kilometers. It varies in width from a few kilometers to more than 60 kilometers. The total area of the deep Hadal zone within the trench is enormous. And with each dive covering only a small area, and with the logistical constraints that make dives possible only under favorable conditions and with considerable preparation and support, the amount of the trench that has been directly observed by humans or their instruments is tiny, a small fraction of a percent of the total area. The rest remains unseen, unmapped in detail, and entirely unknown. What might be living in the parts of the trench we have not yet visited? What might be happening in the kilometers of seafloor between the spots where a submersible has set down and turned on its lights? These are not merely rhetorical questions. Every time a new dive is made into the deep ocean, something unexpected is found. Every time, there is no dive report from the deep sea that does not contain at least one observation that surprises the scientists who made it. The deep ocean has a track record of delivering the unexpected with every visit, which strongly suggests that the inventory of what lives down there and what processes are occurring is far from complete, far from anything close to complete. The Fendouzhe program has made multiple dives into the Mariana Trench, and each one has contributed something new to the picture. New species documented, new behaviors observed. New measurements of chemical and physical parameters that refine the models scientists use to understand how the deep sea system works. New images of terrain that had never been seen before. Each dive is a small window opened into a vast dark room. It illuminates a tiny portion of the interior, and the scientist peers through it and sees something remarkable, and then the window closes again and the darkness returns. Building up a comprehensive understanding of the Mariana Trench from these small windows is the work of decades, if not longer. The scientists who study the deep sea understand this. They operate with a long view, knowing that their individual contributions are pieces of a puzzle so large that no single researcher, or even single research program, can hope to assemble more than a small portion of it in a career. It is science conducted with patience, patient as the deep ocean itself, which has been going about its business for millions of years, and which will continue to do so long after any individual scientist's career is over. This patience is not resignation. It is not an acceptance of ignorance, but an acknowledgement of scale. The researchers who study the Mariana Trench and the broader deep ocean are among the most passionate and driven scientists in any field. They are motivated by the knowledge that what they do not yet know vastly exceeds what they do know, and that this gap represents not a failure but an opportunity. Every unanswered question about the deep ocean is a potential discovery. Every dive into the dark is a chance to see something that nobody has ever seen before. The technology that makes these dives possible has evolved dramatically over the decades since Jacques Piccard and Don Walsh made their historic descent in the Trieste. The development of new materials, new manufacturing techniques, new electronic systems, new camera technologies, and new sampling equipment has transformed what is possible in deep-sea exploration. But the pace of that development and the continued limitations of what current technology can do tells us something important about where we stand in the broader project of exploring and understanding the deep ocean. The fundamental challenge has not changed. You need to get something, a vehicle, instruments, or ideally human observers, to the bottom of the ocean, and you need to keep it functioning under the enormous pressure that exists there. The pressure at the Challenger Deep is approximately 1,100 times atmospheric pressure, and this creates engineering challenges that have not been fully solved even today. The vessels that can currently make the journey are enormously expensive to build and operate, which limits how many of them exist and how frequently they can be deployed. The Fendouzhe is one of only a very small number of full ocean depth crewed submersibles in the world. The infrastructure required to support a deep dive expedition, the support ship, the specialized handling equipment, the crew of engineers and scientists and support personnel is considerable. Even with the most advanced technology currently available, exploration of the Mariana Trench proceeds slowly. Each dive is a major undertaking. The logistics of getting a research ship to the right location, preparing the submersible, conducting the dive safely, and recovering the vehicle and its crew and samples require days of careful work. The number of dives that can be conducted in a single expedition is limited. And each dive, however carefully planned, can survey only a tiny portion of the vast and varied terrain of the trench. This limitation is not simply a matter of time and resources, though those are genuine constraints. It is also a fundamental challenge of exploration in 3 dimensions at extreme scales. The Mariana Trench is not a flat-bottomed canyon with a simple, uniform floor. It is a complex, highly varied terrain, with areas of flat sediment-covered seafloor, rocky outcrops, steep walls, talus slopes of fallen rock, possible cold seep areas, areas of different sediment composition and depth, a diverse landscape as varied and complex as any on land, but without the benefit of aerial or satellite imagery to give us an overview. On land, we can see the terrain from above, from aircraft, from satellites, and use this overview to plan ground surveys. In the deep sea, we have no such overview. Our maps of the deep sea floor, including the Mariana Trench, are constructed from sonar surveys conducted by surface ships, and while these surveys can provide bathymetric data, measurements of depth, they cannot tell us what the seafloor actually looks like up close, what lives there, or what processes are occurring. To truly understand the Mariana Trench, we need both the overview and the close-up view. We need the broad-scale mapping that sonar can provide, and we need the detailed, organism-level observations that only a submersible diving to the bottom can produce. Combining these 2 types of information is a long-term project that is still very much in its early stages. What we have learned so far from the dives that have been made and the samples that have been collected and the measurements that have been taken has already overturned many of our previous assumptions about what the deep ocean is like and what lives there. The discovery of abundant life at the bottom of the Challenger Deep, the documentation of giant amphipods thriving under crushing pressure, the finding of snailfish adapted to live at depths that exceed the ability of any other vertebrate to survive. The revelation of complex biochemical adaptations that allow organisms to function at pressures that would destroy most biological molecules. The demonstration that the deep sea is connected to the surface ocean through the rain of organic material and the dramatic deliveries of whale falls. The disturbing but important finding that human pollution reaches even to the deepest point on Earth. All of this represents genuine, hard-won scientific knowledge. All of it has required enormous effort, technical ingenuity, and physical courage to obtain, and all of it, remarkable as it is, represents only the beginning of what there is to know about the Mariana Trench. The trench is patient. It has been here for tens of millions of years, and it will be here for tens of millions more. The creatures that live at its bottom have been adapting to its conditions for unimaginable spans of time, evolving and diversifying in the dark while we on the surface were not yet evolved to wonder about them. The trench does not need our understanding. It does not require our attention or our exploration. It will go on being what it is whether we study it or not, but we need the trench. We need it not just for the specific scientific knowledge it contains, though that is immensely valuable. We need it for what it tells us about our planet, about the systems that regulate the chemistry of the ocean and thereby the chemistry of the atmosphere and thereby the climate of the Earth, about the breadth and depth in every sense of the biosphere we inhabit, about the limits of life and the extraordinary reach of evolution, about our own place in the planetary system we are part of. The Mariana Trench is not separate from the planet we live on, it is part of it. The deep ocean is not a separate world from the surface world we inhabit. It is the same world, just deeper. The processes occurring at the bottom of the trench, the cycling of carbon, the production and consumption of organic matter, the chemical exchanges between the sediment and the water, the biological communities that live and reproduce and evolve in the dark. All of these processes are part of the same planetary system that includes the surface ocean, the atmosphere, the land, and us. Understanding the Mariana Trench is, in the end, part of understanding the planet. And understanding the planet is not an abstract scientific exercise. It is the most practically important project that science can undertake because it is the foundation of our ability to live on this planet sustainably and to understand how our actions affect the systems that sustain us. The discovery that microplastics reach the bottom of the Challenger Deep is not just an interesting data point about deep-sea chemistry. It is a signal, a clear, unambiguous signal, that the human footprint extends to every corner of the planet, including the deepest and most remote corners. The deep ocean absorbs carbon dioxide from the atmosphere, playing a critical role in moderating the pace of climate change. The organisms living in the deep sea process organic material and recycle nutrients that eventually return to the surface ocean and support the productivity of the photic zone. The deep ocean circulation patterns transport heat and nutrients around the globe, shaping climate on a planetary scale. The deep ocean is not separate from us. We are connected to it and it to us in ways that are only beginning to be fully understood. This is the frame within which the exploration of the Mariana Trench must ultimately be understood, not as an adventure story, though it contains extraordinary adventure, not as a catalogue of strange creatures, though the creatures it contains are genuinely extraordinary, but as the most recent chapter in the long, slow, still ongoing project of understanding the planet we live on, its structure, its systems, its boundaries, its capacities, and its limits. The Mariana Trench represents, in this sense, not just a place but a question. A vast, deep, dark, and immensely complex question that we have only just begun to pose, let alone answer. The Fendouzhe has opened a new window into this question. The footage it has brought back, the samples it has collected, the measurements it has made, the organisms it has documented, all of this has enriched our understanding in ways that were not possible before. But it has also made very clear how much remains unknown, how vast the unanswered territory still is, and how long and difficult the road ahead will be. We are at the beginning of understanding the deep ocean. We are at the earliest stages of a scientific project that will take generations to complete, assuming it can ever be completed at all. The Mariana Trench, which seemed for so long to be simply the deepest hole on Earth, an extreme curiosity, a geological superlative, has turned out to be a living, breathing, dynamically complex ecosystem that is connected to and important for the health of the broader planetary system. The bottom of the world is not empty. It is not barren. It is not the dead and silent void we once imagined. It is alive in ways we are only beginning to understand, with creatures we are only beginning to know, engaged in processes we have only begun to document, playing roles in planetary systems we have only started to appreciate. And there is so much more down there, so much more waiting in the dark, patient as the deep ocean itself, for us to find the technology and the will and the wisdom to go and look. This is the Mariana Trench. This is the deepest place on Earth. And the story of what lives there, the true, full story, is still being written, one dive at a time, in the dark at the bottom of the world. The sediment at the bottom of the Mariana Trench is not merely a passive substrate on which the drama of the deep-sea ecosystem unfolds. It is itself a living, active, chemically dynamic system, a record and a participant in the processes of the deep ocean. Understanding what the sediment contains and how it functions is one of the central tasks of deep-sea research, and the samples collected by the Fendouzhe and other deep-sea missions have provided unprecedented insight into a world that is, quite literally, beneath the bottom of the world. When organic particles settle out of the water column and reach the seafloor of the Mariana Trench, they do not simply sit on the surface of the sediment indefinitely. They are processed. They are consumed by bacteria and other microorganisms. They are mixed into the sediment by the burrowing and feeding activities of benthic invertebrates, the organisms that live in and on the sediment. They are chemically transformed by a series of reactions that release nutrients back into the water and produce gases that can escape upward through the sediment and into the overlying water column. This processing of organic material in the sediment, what marine scientists call benthic respiration or sediment diagenesis, is a critical part of the ocean's broader biogeochemical cycles. The deep sea floor, including the Hadal sediments of the Mariana Trench, is a major site of carbon remineralization, the conversion of organic carbon, the carbon stored in biological molecules, back into inorganic carbon, carbon dioxide, bicarbonate, that can be released back into the water. This process closes the biological pump, the cycle by which carbon dioxide is fixed into organic matter by photosynthesis at the surface, and then transported to the deep ocean through sinking particles, where it is eventually respired back to inorganic form. The biological pump and the remineralization that occurs in the deep sea sediment are critical regulators of the ocean's capacity to absorb carbon dioxide from the atmosphere. The efficiency of the biological pump, how much of the organic carbon fixed at the surface ultimately reaches the deep sea and is stored there for long periods, determines, in part, how effective the ocean is as a sink for atmospheric carbon dioxide. Understanding what happens to organic carbon when it reaches the deep sea floor, including the floor of the Mariana Trench, is therefore not just an academic curiosity. It is directly relevant to understanding the carbon cycle, and by extension, the climate system. The microbial communities in the Hadal sediment of the Mariana Trench play a particularly important role in this process. These communities, which are dominated by bacteria and archaea, with smaller contributions from more complex microorganisms, are responsible for the vast majority of the biological processing of organic material at these depths. They break down complex organic molecules into simpler compounds, respire carbon dioxide, cycle nitrogen and sulfur and other elements through a series of transformation reactions, and collectively drive the chemical transformation of the sediment. What has surprised researchers who have studied the microbial communities in Hadal sediments is both their diversity and their activity levels. The assumption prior to direct investigation was that microbial activity in the deep, cold, nutrient-poor Hadal environment would be extremely low, that the microbes would be present but barely metabolically active, ticking over at the slowest possible rate compatible with survival. Instead, what has been found is that the microbial communities in Hadal sediments can be quite active, and that their activity is enhanced by the concentration of organic material that results from the funneling effect of the trench geometry. The very deepest parts of the trench floor, which receive the highest concentrations of settling organic material, also tend to host the most active and diverse microbial communities. This finding has important implications for our understanding of the deep ocean's role in carbon cycling. If Hadal sediments are more biologically active than previously thought, if more organic carbon is being respired back to carbon dioxide at these depths than our models predicted, then our estimates of how much carbon is ultimately stored in the deep ocean may need to be revised. The deep ocean is not simply a passive reservoir for organic carbon. It is an active processing environment, and the biology of that processing, including the biology of the Mariana Trench, matters for our understanding of global carbon cycling. The samples brought back from the trench by the Fendouzhe and other missions have allowed scientists to begin characterizing the microbial communities in the Hadal sediment in detail. Using modern molecular techniques, sequencing the genetic material extracted from sediment samples to identify the organisms present, researchers have found that Hadal sediments contain a diverse and in many ways unique community of microorganisms. Many of the species found in Hadal sediments are not found in shallower sediment environments, suggesting that the extreme conditions of the Hadal zone have driven the evolution of a distinct microbial fauna adapted specifically to life under crushing pressure in the cold and under the specific chemical conditions of the Hadal sediment environment. Some of these microorganisms are of particular scientific interest because of the biochemical pathways they use. Some perform what is called anaerobic respiration, They use compounds other than oxygen as the terminal electron acceptor in their respiratory reactions. In the absence of oxygen, which can become depleted in deep-sea sediments, microorganisms can use sulfate, nitrate, iron, or other compounds to accept electrons in their respiratory chains. These different types of respiration drive different chemical transformation reactions in the sediment, producing different products and transforming the sediment chemistry in different ways. Understanding which types of respiration are dominant in Hadal sediments and how this changes with depth in the sediment and with location in the trench is a key part of understanding the geochemistry of the deep sea floor. The study of these microbial communities and their metabolic activities is a relatively young field, made possible by the development of modern molecular biology techniques and by the availability of sediment samples collected from Hadal depths. It is a field that is advancing rapidly, with each new expedition and each new set of samples adding to the picture, but it is also a field that is still far from complete. The diversity of microbial life in the deep ocean is enormous, far greater than what has been cataloged so far, and understanding the ecological roles of the different microbial taxa and how they interact with each other and with the physical and chemical environment is a project that will occupy researchers for many years to come. As extraordinary as the biology of the Mariana Trench is, and as important as the biochemical adaptations of deep-sea organisms are, there is another dimension of the trench ecosystem that deserves careful attention: the physical dynamics of the water column above the trench floor. The Mariana Trench is not just a hole in the seafloor. It is an oceanographic feature that interacts with the broader water column and circulation of the Pacific Ocean in ways that affect the biology and chemistry of the trench environment. Water in the deep ocean is not still. Despite the popular image of the deep sea as a calm, motionless world, an image reinforced by the apparent tranquility seen in submersible footage, the deep ocean has its own currents and circulation patterns, driven by differences in temperature and salinity and by the movement of water masses on a global scale. The thermohaline circulation, the global system of ocean currents driven by density differences in the water, reaches to the very bottom of the ocean. Deep water masses formed at high latitudes where surface water becomes cold and dense enough to sink, flow along the ocean floor and eventually fill all the world's ocean basins, including the deepest trenches. The Mariana Trench receives water from the general circulation of the Pacific Basin, but the trench geometry also creates local circulation effects. Water can be trapped in the trench, particularly in the deepest parts, where the steep walls prevent horizontal water movement. Temperature and chemical measurements in the deepest parts of the trench suggest that the water there is relatively isolated from the overlying water column. It is not replaced as rapidly as water in shallower parts of the ocean, and it can develop distinctive chemical characteristics as a result of the biological and chemical processes occurring within the trench. This isolation has implications for the biology of the trench. If water in the deep trench is relatively isolated, then chemical signals, the traces of decaying organic material, the products of microbial metabolism, the molecules released by living organisms, can accumulate to higher concentrations than they would in a more well-mixed environment. This can affect which organisms are attracted to the trench environment and how they navigate within it. The concentration of chemical signals in the water above the trench floor may be one of the reasons that deep-sea scavengers are able to locate food sources as quickly as they do. The chemical gradients in the relatively isolated water of the trench bottom may be stronger and more persistent than they would be in a more open, well-mixed environment. The physical dynamics of the water column also affect how organic material reaches the trench floor. The descent of organic particles through the water column is not a simple, undifferentiated process. Particles of different sizes, shapes, and densities sink at different rates. Some particles are consumed by organisms in the water column before they reach the bottom. Some aggregate with other particles as they sink, forming larger clumps that sink more rapidly. Some are remineralized by bacteria during their descent, reducing the amount of organic carbon that ultimately reaches the floor. The specific characteristics of the water column above the Mariana Trench, its temperature structure, its chemistry, its biological communities, determine how much organic material ultimately reaches the trench floor and in what form. Understanding all of these factors, the physical dynamics of the water column, the settling of organic material, the biology of the mid-water communities through which the material passes, the chemistry of the deep trench water, and the biology and chemistry of the sediment at the bottom, is what will eventually allow scientists to build a comprehensive, quantitative model of how the Mariana Trench functions as an ecosystem and how it is connected to the broader ocean system. That model does not yet exist. We have pieces of it, measurements of depth, temperature, chemistry, observations of organisms, samples of sediment, footage of the seafloor, but assembling these pieces into a coherent, quantitative whole is the work that remains to be done. It is work that requires repeated visits to the trench, each one adding new data to the picture. It requires the continued development of new instruments and sampling techniques capable of making measurements that current technology cannot. It requires collaboration between scientists of many different disciplines, biologists, chemists, physicists, geologists, oceanographers, each bringing a different perspective and a different set of tools to the shared project of understanding the deep. And it requires, perhaps most fundamentally, the willingness to sit with what we do not know. To acknowledge that the picture we have so far is incomplete, that the questions we have answered have generated many more questions than they resolved, and that the deep ocean, including the Mariana Trench, is vastly more complex and more interesting than we imagined when we first began to study it. The Mariana Trench has been teaching us this lesson since 1875, when the crew of HMS Challenger lowered a weighted line over the side of their ship and found that it kept going down and down and down farther than anything they had ever measured before. In the century and a half since then, everything we have learned about the trench has confirmed and deepened this lesson, that the world is larger, more complex, more richly alive, and more mysterious than we tend to assume. The deep is not empty. The deep is full of life, full of process, full of connection, full of meaning, and we have only just begun to understand it, to truly appreciate what the exploration of the Mariana Trench means and why it matters. It helps to think about it in the context of what we know about the history of life on Earth. Life on this planet is ancient. The earliest evidence of microbial life dates back more than 3.5 billion years, to a time when the Earth was a very different place, with a different atmosphere, different oceans, different continents, and a sun that burned somewhat cooler than it does today. For the first 2 billion years or so of this long history, life was exclusively microbial. The only living things on Earth were single-celled organisms, microscopic in size, engaging in a variety of metabolic strategies that we are still working to fully catalog and understand. These ancient microbes lived in a world that in many ways resembled the deep ocean more than it resembled the surface world we are familiar with. The early Earth had little or no oxygen in its atmosphere. Photosynthesis, the oxygen-producing kind that created the oxygen atmosphere we breathe today, had not yet evolved or was just beginning to emerge. Many of the metabolic processes that were most important in the early biosphere were chemosynthetic. They relied on chemical energy rather than sunlight. And many of the environments where life thrived in the early biosphere were hot, dark, chemically extreme environments somewhat analogous to the hydrothermal vents and cold seeps of the modern deep ocean. This deep antiquity of chemosynthesis-based life is not lost on the scientists who study deep-sea ecosystems. There is a school of thought not universally accepted, but taken seriously, that the deep-sea hydrothermal vent environment may be where life on Earth first originated. The chemistry of hydrothermal vents provides the raw ingredients and the energy necessary for the synthesis of complex organic molecules from simple inorganic precursors. The mineral surfaces of vent chimneys can catalyze chemical reactions that might otherwise be too slow to be significant. The warm, chemically energetic environment of a hydrothermal vent has features that could, in principle, have supported the first self-replicating chemical systems, the proto-life that preceded the emergence of true cellular life as we now understand it. Whether or not life actually originated at hydrothermal vents, the connection between the chemistry of the early Earth and the chemistry of the modern deep sea is real. The metabolic pathways used by chemosynthetic microorganisms at deep-sea vents and in Hadal sediments are ancient. They predate the evolution of photosynthesis by a vast span of time. They represent the original energy strategies of life on Earth, preserved in the modern biosphere in the places that are still most similar, in terms of their chemistry and their energy landscape, to the conditions that prevailed when life first emerged. The Mariana Trench is, in this sense, a window into the deep past of life on Earth. The organisms living at its bottom, the chemosynthetic microbes, the pressure-adapted invertebrates, the strange and slowly living fish of the Hadal zone, are not just extreme curiosities. They are the modern representatives of ancient lineages that have been finding ways to live in dark, pressurized, chemically extreme environments for a very long time. They are living archives of the Earth's biological history, carrying within their cells and their genomes the traces of billions of years of adaptation to conditions that are as extreme as anything our planet has to offer. This perspective, the deep time perspective, the view of the Mariana Trench ecosystem as a living relic of ancient Earth conditions, is one that researchers increasingly bring to their work. When they study the biochemistry of pressure-adapted proteins in Hadal organisms, they are not just learning how those specific organisms survive. They are learning about the relationship between life and its physical environment in the most fundamental terms. When they sequence the genomes of archaea living in Hadal sediments, they are reading documents that encode billions of years of evolutionary history. When they observe the cold seep communities clustered around areas of chemosynthetic activity in the trench, they are seeing something that may be continuous with the very oldest communities of life on Earth. The Mariana Trench is ancient. It has been collecting life for as long as it has existed, tens of millions of years. The organisms living there have had all that time to adapt, to diversify, to evolve specialized traits for their specific circumstances. Some of the lineages represented in the Hadal fauna are among the oldest continuous lineages in the biosphere. They have persisted through geological ages, through mass extinctions, through the rise and fall of entire groups of organisms that dominated the surface ocean and then disappeared, while the deep ocean community has continued slowly in the dark. This persistence is itself remarkable. The deep ocean has been one of the most stable environments on Earth over geological time. While the surface of the planet has been buffeted by changes in temperature, in the composition of the atmosphere, in the geography of the continents, in the productivity of the surface ocean, while mass extinctions have repeatedly devastated the biodiversity of the surface world, the deep ocean has remained relatively stable. Cold, dark, pressurized, and fed by the slow rain of material from above, the deep ocean has provided a refuge for life through events that wiped out the majority of species living in other environments. This stability and persistence have their own implications for what we might find in the Mariana Trench as exploration continues. The deep ocean, including the Hadal zone, may harbor lineages of organisms that are, in evolutionary terms, living fossils. Organisms whose ancestors have not changed fundamentally in tens or even hundreds of millions of years, because the environment they live in has not changed enough to drive rapid evolutionary change. Discovering and studying these organisms could provide extraordinary insights into the history of life on Earth, into the biological conditions of ancient oceans, into the evolution of fundamental biochemical processes, into the relationships between ancient and modern organism groups. We do not yet know what living fossils the Mariana Trench may be concealing in its unmapped terrain. We do not yet know what ancient lineages persist in the sediment of the Challenger Deep, invisible to our current methods of sampling and analysis. But the possibility that they exist, that the deepest parts of the ocean are reservoirs of ancient, barely changed life, is one of the most exciting prospects for future deep-sea research. As we draw toward the end of this first part of our exploration, our attempt to begin understanding what the Mariana Trench is, what it contains, and why it matters, it is worth pausing to reflect on what we have covered and what it adds up to. We have traveled conceptually from the surface of the Pacific Ocean down through nearly 11 kilometers of water to the floor of the deepest place on Earth. We have traced the geological history of the trench, the 180 million years of subduction that created it. We have followed the history of human attempts to reach it, from the first soundings made by HMS Challenger through the extraordinary dives of the Trieste and the deep-sea Challenger to the current systematic exploration conducted by the Fendouzhe. We have met the organisms that live at its bottom, the giant amphipods, the Hadal snailfish, the chemosynthetic microbes, the strange and diverse invertebrates that populate a world of crushing pressure and total darkness. We have examined how these organisms survive through the molecular adaptations that allow them to function at pressures that would destroy any unmodified biological system. We have explored how the ecosystem functions, how energy and nutrients move through it, how the connection to the surface ocean sustains it, how whale falls transform it temporarily into places of extraordinary productivity. We have confronted the disturbing evidence of human pollution reaching even to the deepest point on Earth, and we have situated all of this within the broader context of deep time, planetary systems, and the fundamental question of what life is and what it can do. What does it all add up to? It adds up to a revolution in our understanding of the biosphere, a recognition that the world of life is vastly larger, more complex, and more resilient than we ever imagined. That life does not simply occupy the comfortable, sunlit, moderate environments we typically associate with biological flourishing. Life penetrates into the most extreme conditions the planet offers. It adapts, it persists, it evolves, it finds a way. And in finding its way into the deepest trench of the deepest ocean, life has built a world, a real, functioning, complex, ancient world that we are only now beginning to see. The Mariana Trench is not a curiosity at the edge of the biosphere. It is the biosphere's most extreme expression. It is the place where life has been pushed to its absolute limit and has responded not by failing but by adapting in ways that are among the most remarkable biological achievements this planet has produced. Understanding it is not an optional extra for planetary science. It is essential, and the work is far from done. The vast majority of the trench remains unexplored. The sediment harbors communities and processes we have not yet characterized. The water column above the trench floor teems with organisms we have not yet fully identified. The geological and chemical processes occurring at the deepest point on Earth are not yet fully understood. The connections between the Hadal ecosystem and the broader ocean, and through the ocean to the climate and the carbon cycle and the planetary systems that sustain all life on Earth, are not yet quantified. There is much, much more to learn. The path forward leads downward into the dark, into the deep, into the patient and ancient world at the bottom of the ocean. And every step down that path, every dive, every sample, every measurement brings us closer to understanding not just the Mariana Trench, but the planet we call home. The second part of this journey will take us deeper still into the specific findings of the most recent expeditions, into the cutting edge of what science is now discovering about the deep sea, into the future of deep ocean exploration and what it might reveal. The bottom of the world is still giving up its secrets, slowly, one dive at a time. And the story of what lies there, the true, full, extraordinary story, is still unfolding. We have only just begun. We left the first part of this journey at the edge of understanding. We had descended, conceptually and scientifically, through nearly 11 kilometers of water to the floor of the deepest place on Earth. We had met the organisms living there, creatures that should not exist by any comfortable reckoning of what biology is capable of, and yet do exist, thriving in conditions that would kill almost anything else alive. We had traced the connections between the deep trench ecosystem and the surface world above it, the rain of organic material, the dramatic arrivals of whale carcasses, the microplastics that drift down from a world of human activity that the organisms at the bottom of the Challenger deep have never seen and never will. And we had sat with the uncomfortable reality that despite all of this, despite the extraordinary scientific progress of recent decades, despite the engineering achievements that have made it possible to visit the deepest point on Earth multiple times, the Mariana Trench remains, in its vast majority, completely unknown. Now we go deeper, not in terms of physical depth, we are already at the bottom, but in terms of understanding. In this second part of our journey, we are going to follow the most recent scientific expeditions into the trench in greater detail. We are going to look closely at specific discoveries that have fundamentally changed the way scientists think about the deep ocean. We are going to examine the organisms of the Hadal zone with a level of biological precision that reveals just how extraordinary their adaptations really are. We are going to look at what the trench can tell us about the planet as a whole, about its climate, its carbon cycle, its geological history. We are going to peer into the future of deep-sea exploration and ask what the next generation of technology might be able to reveal. And we are going to grapple with the philosophical implications of everything we have found, with what it means that life exists at the very bottom of the world, and what that existence tells us about life's fundamental nature. The Mariana Trench is not done with its revelations. In fact, the most important ones may still be waiting, buried in the sediment of the Challenger deep or swimming in the dark water above its floor, patient and unhurried, waiting for us to develop the tools to find them. Let us go and look. Every successful scientific expedition to the Mariana Trench begins the same way, with preparation. With months of planning, instrument calibration, logistical coordination, and the kind of meticulous attention to detail that is absolutely non-negotiable when you're building a system intended to keep human beings alive at a pressure of over 1,000 atmospheres. The scientists and engineers who work on deep-sea submersibles understand this at a visceral level. There is no margin for error at the bottom of the Challenger deep. A flaw in a seal, a mistake in a calculation, a component that performs perfectly in testing but fails under the actual conditions at extreme depth, any of these things can be fatal. The engineering culture of deep-sea exploration is, of necessity, a culture of extreme caution and redundancy. Everything that can fail must be identified. Everything critical must have a backup. And even with all of that, there is always the knowledge that the deep ocean is an environment of such severity that uncertainty can never be fully eliminated. The Fendouzhe submersible embodies this culture of meticulous engineering in its every component. The pressure hull, the sphere in which the crew rides, is made from a high-strength titanium alloy, chosen for its combination of strength, low density, and resistance to corrosion. The sphere is machined to extremely tight tolerances to ensure that it is as close to perfectly round as manufacturing can achieve. Deviations from perfect roundness create stress concentrations under pressure that can propagate into cracks, so the machining precision required is extraordinary, measured in fractions of a millimeter across a sphere nearly 2 meters in diameter. The viewports of the Fendouzhe deserve particular attention, because they represent one of the most challenging engineering problems in the entire vehicle. A viewport is, by definition, a hole in the pressure vessel, a place where the integrity of the hull is interrupted to allow the crew to see outside. Under the pressure of the Challenger deep, a viewport that is not perfectly designed and manufactured will fail, and failure will be catastrophic. The viewports of full ocean depth submersibles are made of a transparent material, typically a special formulation of acrylic, that must be thick enough to withstand the pressure without cracking or deforming. The design of the viewport seal, the interface between the transparent acrylic cone and the metal hull, is particularly critical. Under pressure, the acrylic actually compresses slightly, and this compression must be accounted for in the seal design. If the seal is too tight when the viewport is at surface pressure, it will prevent the necessary compression and may crack the acrylic under depth pressure. If it is too loose, it will leak. Getting this balance right requires both precise calculation and extensive testing. The external systems of the Fendouzhe, the thrusters, the manipulator arms, the camera systems, the sampling equipment, all present their own engineering challenges. Electric motors and thrusters must be designed to operate in seawater at extreme pressure, either by being pressure compensated, filled with oil at the same pressure as the surrounding water, so that there is no pressure differential across the housing, or by being rated for the full external pressure with appropriate housing design. Cameras must be housed in pressure-tolerant enclosures with optical windows that do not distort the image. Lights must be powerful enough to illuminate the dark seafloor adequately while consuming minimal power from the submersible's battery system, which is the only power source available during a dive that may last the better part of a day. The battery system itself is a marvel of engineering. The energy required to power the submersible's thrusters, lights, instruments, life support, and communications systems over a dive of many hours is substantial. The batteries must store this energy compactly and reliably, and they must do so in a form that can withstand the pressure of the deep ocean. Lithium-ion batteries, which offer the best energy density of currently available battery technologies, require careful pressure compensation and thermal management. Their performance at the low temperatures of the deep sea must be characterized and accounted for in the vehicle's power budget. All of this engineering effort, the years of design, testing, manufacturing, and refinement that went into building the Fendouzhe, converges on a single purpose: enabling human beings to descend to the deepest place on Earth and spend enough time there to make meaningful scientific observations. When the submersible is ready, when the support ship has been positioned over the Challenger deep, when the weather is favorable and the sea state manageable, the hatch is closed, the pre-dive checklists are completed, and the crane lowers the submersible into the water. The crew, sealed inside their titanium sphere with hours of oxygen and a full suite of instruments, watches through the viewports as the surface world, the blue sky, the white ship, the choppy surface of the Pacific recedes above them and the darkness begins to close in. The descent to the Challenger deep takes several hours. For the first few hundred meters, there is still light, the blue light of the tropical Pacific filtering through the water above. The crew can see the water column through the viewports, can watch for the occasional passing fish or jellyfish or other organism visible in the light. But as the submersible sinks below a few hundred meters, the light fades. By 500 meters, it is effectively gone. Below that depth, the only illumination is the bioluminescence, the living light produced by organisms in the mesopelagic and bathypelagic zones, which flash and pulse and drift through the water in patterns that are beautiful and completely alien. The bioluminescence of the midwater zone is itself a remarkable phenomenon that deserves more attention than it typically receives in discussions of deep-sea biology. Bioluminescence, the production of light by living organisms through chemical reactions, is extraordinarily common in the deep ocean. Estimates suggest that the majority of organisms living in the midwater zone, the part of the ocean between a few hundred and a few thousand meters depth, are capable of producing light. The purposes of this light are varied: attracting prey, repelling predators, communicating with potential mates, providing camouflage through counterillumination, the production of light on the underside of an organism to match the faint downwelling light from above, making the organism less visible to predators looking up from below. The bioluminescent light show visible from the viewport of a descending submersible is a constant reminder that the deep ocean, dark as it is from our surface perspective, is not a world without light. It is a world with a completely different kind of light generated by biology rather than by the sun, used for biological purposes, flickering and drifting through the water in ways that are governed by the ecological and behavioral needs of the organisms producing it. This biological light fills the midwater zone with a diffuse, intermittent, multi-dimensional illumination that is utterly different from anything on the surface but which represents, for the organisms living there, a complex and important part of their sensory environment. As the Fendouzhe continues its descent, passing through the midwater zone and into the deep sea, the bioluminescence gradually becomes sparser. The density of organisms decreases with depth, and with it the frequency of bioluminescent flashes. By the time the submersible is approaching the bottom of the trench, descending through the deepest kilometers toward the floor of the Challenger deep, the water outside the viewports is essentially completely dark, utterly, profoundly, absolutely dark. Not the darkness of a room with the lights off, where there is always some faint residual glow from outside. Not the darkness of a cave, where a sufficient weight often reveals some faint bioluminescence or the glow of minerals. The darkness of the deep Mariana Trench is total in a way that very few people have ever experienced. It is the darkness of a place where no photon of light has penetrated in living memory of the universe, where even the faint, distant echo of the surface world's light cannot reach. And then, after hours of this descent through absolute darkness, the lights of the Fendouzhe reach the seafloor, and in an instant the world changes. The sediment of the Challenger deep, illuminated for the first time by the powerful LEDs of the submersible, glows white in the darkness. It is fine-grained and soft-looking, disturbed by the gentle currents of the bottom water into subtle ripples and ridges that speak of a dynamic environment despite the apparent stillness. And within that illuminated circle, within the narrow cone of visibility that the submersible's lights provide, there is life. The scientific program of the Fendouzhe dives in the Mariana Trench was designed to answer specific questions, but like all good science, it generated as many new questions as it resolved. The principal objectives were to characterize the biology, chemistry, and geology of the Hadal zone in the Mariana Trench with a level of detail and systematic coverage that had not previously been achieved. This required not just single dramatic dives but multiple dives to multiple locations, each building on the findings of the previous one, each contributing data points to an increasingly rich and detailed picture of the deep trench environment. One of the earliest significant observations made during the Fendouzhe dives was the sheer abundance of life visible on the seafloor. Previous deep-sea expeditions, using cameras lowered from surface ships on cables, had provided glimpses of the deep-sea fauna, photographs of amphipods and occasional fish, images of sediment with trails and burrows that testified to the presence of organisms, even when the organisms themselves were not visible. But the Fendouzhe, as a maneuverable vehicle capable of approaching the seafloor slowly and carefully, provided a very different quality of observation. For the first time, it was possible to watch the organisms of the Challenger deep going about their normal activities, to see how they moved, how they fed, how they responded to the presence of the submersible, and how they interacted with each other and with their environment. What the crew and cameras observed was a level of biological activity that consistently surprised even experienced deep-sea biologists. The amphipods, those large, swarm-forming crustaceans that are among the most characteristic animals of the Hadal zone, were observed in quantities that spoke to a healthy, abundant population. They were seen feeding on organic material in the sediment, swimming through the water column above the seafloor, and aggregating in large numbers around any source of food that became available. Their behavior was energetic and purposeful, not the sluggish, barely functioning organisms one might expect in an extreme environment, but active, effective, ecologically competent animals clearly well suited to their environment. The snailfish observed during the dives were perhaps even more striking in their behavior than in their mere presence. These animals, the deepest living fish ever recorded, were seen swimming actively through the water just above the seafloor, moving with apparent ease and showing behaviors clearly oriented toward finding and consuming prey. They were observed feeding on amphipods, demonstrating that the relationship between the fish and the amphipod population is one of predator and prey, that the snailfish sit at a higher trophic level than the amphipods, consuming them and incorporating their biomass into the vertebrate food web of the Hadal zone. This was not entirely surprising. Snailfish in other environments are known to be active predators, but seeing it directly, in the deep Mariana Trench, under the lights of a submersible at the bottom of the world, was a different thing from simply inferring it from theoretical considerations. The behavior of the snailfish around the submersible itself was informative. Unlike many deep-sea animals which flee from the lights and noise of approaching vehicles, the snailfish observed during the Fendouzhe dives often showed relatively little fear response to the submersible. They approached it, investigated it, and went about their feeding activities in its presence. This behavior is consistent with what is known about the general principle that deep-sea animals tend to be less skittish around artificial light sources than their shallow water relatives, perhaps because they have not evolved strong responses to sudden light, which is not a feature of their natural environment, or perhaps because the Fendouzhe, with its relatively quiet electric thrusters and gentle movements, does not trigger the same threat responses that a fast-moving predator might. Beyond the biology of individual organisms, the Fendouzhe dives generated important observations about the physical structure of the seafloor at different locations within the trench. The terrain at the bottom of the Mariana Trench is not uniform. Different areas show dramatically different character. Some areas are flat plains of soft sediment, while others are marked by rocky outcrops, steep slopes, and other features that reflect the complex geological history of the trench floor. The sediment itself varies. In some areas it is thick and soft, in others thin and interrupted by exposures of harder material beneath. These variations in terrain correspond to variations in the biological communities. On soft sediment plains, where organic material accumulates more effectively, the fauna tends to be dominated by the soft-bodied organisms that burrow into sediment and by the amphipods and other mobile organisms that forage across the sediment surface. On rocky outcrops where the substrate is harder and less accessible for burrowing, different organisms attach to the rock surface. Various types of worms, sponges, and other sessile or semi-sessile organisms that can find purchase on the hard substrate. The ecological diversity of the trench bottom is thus partly a function of geological diversity, of the varied substrates that the complex terrain of the trench provides. Mapping this terrain, understanding where the different substrate types occur, how the topography varies across the length and width of the trench, where the deepest points are, and how they compare in terms of biology and chemistry, is an ongoing project that the Fendouzhe program has significantly advanced. The combination of sonar data from the support ship and direct visual observations from the submersible is gradually building a picture of the trench floor that is far more detailed and accurate than anything previously available. Among the most scientifically significant aspects of the Fendouzhe program's findings are those related to the detailed biology and biochemistry of the organisms collected during the dives. Bringing organisms back from the Challenger deep for laboratory analysis is technically challenging. Many deep-sea organisms are sensitive to the changes in pressure and temperature that occur during the ascent and die or are severely damaged before they can be analyzed. But techniques have been developed to address this challenge, including pressure-maintaining collection containers that keep specimens at or near the pressure of their natural environment during the ascent, and rapid analysis techniques that can characterize biological samples quickly enough to capture information that might otherwise be lost. The organisms collected from the Mariana Trench and analyzed in surface laboratories have yielded a wealth of biochemical information that is changing our understanding of how life functions at extreme pressures. The work on trimethylamine oxide in snailfish has already been mentioned, but it is worth examining in more detail. because the findings go beyond simply confirming that TMAO is present in deep-sea fish. Researchers have found that the concentration of TMAO in snailfish tissues increases predictably with the depth at which the fish lives, following a linear relationship that suggests this molecule is actively regulated in response to the pressure the fish experiences. The deeper the fish lives, the more TMAO it needs to maintain its protein function, and its biology adjusts accordingly. This finding has implications that extend far beyond the snailfish. If TMAO concentration is a reliable indicator of the pressure environment in which a fish lives, it provides a potential tool for reconstructing the depth history of fish populations, for understanding whether the ancestors of current deep-sea fish lived at similar depths or colonized the deep ocean from shallower environments. It may also provide insights into the evolutionary history of pressure adaptation. When did deep-sea lineages first develop the TMAO-based pressure adaptation, and how has this adaptation refined itself over evolutionary time? The pressure adaptation story in deep-sea organisms is also providing insights into a general principle in molecular biology, the relationship between molecular stability and function. Life requires molecules that are stable enough to maintain their structure and carry out their functions, but not so stable that they become rigid and incapable of the conformational changes required for enzymatic activity. In shallow water organisms, this balance is struck under atmospheric pressure conditions. In deep-sea organisms, the balance must be maintained under conditions of extreme pressure. Understanding how deep-sea organisms achieve this balance through TMAO stabilization, through membrane lipid composition, through specific mutations in protein sequences that modify their pressure sensitivity, is teaching biochemists fundamental things about the physical chemistry of biological molecules and how the balance between stability and flexibility is achieved in different environments. The genome sequencing of deep-sea organisms is providing another window into these adaptations. When the full genome sequences of Hadal organisms are compared with the sequences of their shallow water relatives, the differences in the protein coding regions of the genome can sometimes be traced to specific adaptations for pressure. Mutations that alter the amino acid sequences of proteins, particularly enzymes, can affect how those proteins respond to pressure, and in deep-sea organisms, natural selection has apparently favored mutations that reduce pressure sensitivity. Identifying these specific mutations and understanding their biochemical effects is a growing area of research that is beginning to provide a molecular picture of how pressure adaptation works at the genetic level. The amphipods of the Mariana Trench have been particularly well studied in this regard, in part because they are abundant and relatively easily collected in large numbers, providing enough material for detailed biochemical and genomic analysis. The giant amphipods of the Hadal zone turn out to be biochemically remarkable in multiple ways. Their enzyme systems have been modified to function efficiently under pressure. Their cuticle, the external skeleton made of chitin and proteins that covers the body of crustaceans, has a chemical composition that differs from that of shallow water relatives in ways that may reflect pressure adaptation. Their lipid composition shows the same shift toward unsaturated fatty acids in cell membranes that is seen in other deep-sea organisms, maintaining membrane fluidity under high pressure. But perhaps the most unexpected finding about the Hadal amphipods concerns their chemical ecology, specifically the chemicals they accumulate in their tissues and where those chemicals come from. Analysis of the fatty acid composition of Hadal amphipods has revealed the presence of specific fatty acids that are found in the surface ocean, but are relatively uncommon in the deep-sea fatty acids produced by photosynthetic organisms in the sunlit zone. The presence of these compounds in the tissues of amphipods living at the bottom of the Mariana Trench, far from any photosynthesis, is direct chemical evidence of the connection between the surface and the deep. These amphipods are eating organic material that originated in the photosynthetic surface ocean, and the chemical signature of that origin is preserved in their body tissues. This kind of chemical tracer approach, using the specific chemical composition of deep-sea organisms as a record of what they have eaten and where their energy ultimately came from, is one of the powerful analytical tools that modern deep-sea research has developed. It allows scientists to trace the flow of energy and nutrients through the deep-sea food web with a precision that was not possible when researchers were limited to direct observation. Combined with isotopic analyses, which use the ratios of stable isotopes of carbon, nitrogen, and other elements to trace food web connections, these chemical approaches are providing a detailed and quantitative picture of how the deep-sea ecosystem is organized and what drives it. The question of how the deep-sea ecosystem responds to change over time is one of the most important and least well-understood aspects of deep-sea biology. On the surface, ecosystems respond to seasonal changes in temperature, light, and the availability of resources. They pulse with the rhythms of the seasons, ramping up productivity in favorable periods and contracting in unfavorable ones. In the deep sea, the absence of light means there is no direct seasonal signal of the same kind that drives surface ecosystems. But there is an indirect seasonal signal, transmitted through the water column by the sinking of organic material from above. In the surface ocean, primary production, the photosynthetic activity that forms the base of the marine food web, is strongly seasonal in many parts of the world. The spring bloom, which occurs in temperate and subpolar regions as day length increases and stratification of the water column develops, represents a massive pulse of photosynthetic productivity that produces an enormous quantity of organic matter. Much of this organic matter is consumed in the surface waters, but a portion sinks, eventually reaching the deep sea. This seasonal pulse of sinking organic material, delayed by the time it takes to sink through thousands of meters of water, dispersed by the currents it passes through, reduced in quantity by consumption during descent, but still detectable, reaches the deep sea floor weeks to months after the surface bloom occurs. The deep sea ecosystem does respond to this pulse. Studies at deep-sea observatories, instrumented platforms on the seafloor that make continuous measurements over years, have detected seasonal cycles in the flux of organic material to the seafloor and corresponding seasonal changes in the biological activity of the deep-sea community. These cycles are damped and delayed compared to the surface signal, but they are real. The deep sea is not completely insulated from the seasonal rhythms of the surface. Whether this seasonal response extends to the Hadal zone of the Mariana Trench is a question that has been difficult to answer, because the monitoring of deep-sea conditions over time requires long-term instrumentation that is technologically challenging and expensive to deploy in the trench environment. The extreme pressure, the remoteness, and the logistical difficulty of maintaining instrumentation at Hadal depth have meant that continuous time series data from the trench is very limited. Most of what we know about the Mariana Trench comes from single-visit expeditions, snapshots of the ecosystem at a particular moment, rather than a continuous record of how it changes over time. This is one of the most significant gaps in our current understanding of the Hadal ecosystem. A single expedition tells us what the trench looks like on the day of the dive. It does not tell us how representative that observation is of the typical state of the ecosystem, how much the conditions vary from season to season or from year to year, or how the ecosystem responds to longer-term changes in the surface ocean above it. To answer these questions, we need sustained monitoring instruments left in place at the bottom of the trench for months or years, continuously recording the conditions there and transmitting that data back to the surface. The deployment of such instruments in the Mariana Trench presents formidable engineering challenges. The pressure at the bottom of the Challenger Deep will destroy conventional oceanographic instruments that have not been specifically engineered for Hadal conditions. The instruments must be designed to record and store data over extended periods without requiring maintenance. There is no possibility of sending a technician down to fix a broken instrument. The power supply must last for the entire deployment period without recharging. And at the end of the deployment, the instrument must be able to return to the surface, typically by releasing a ballast weight and floating up, reliably enough that the data it has collected can be recovered. Despite these challenges, Hadal lander deployments, sending instrumented vehicles to the bottom of the trench without a crew, leaving them there for an extended period, and then recovering them, have become an important part of the deep-sea research toolkit. These landers can carry cameras to record biological activity over time, water chemistry sensors to monitor changes in the chemical environment, current meters to measure water movement at the bottom of the trench, and sediment traps to collect settling particles and characterize the flux of material arriving at the seafloor. The data from these landers, combined with the direct observations from crewed submersible dives, is beginning to build a more complete and dynamic picture of the Mariana Trench ecosystem. What limited time series data exists from the deep Mariana Trench and surrounding Hadal environments suggests that the biology of the trench bottom does fluctuate over time, likely in response to the variable supply of organic material from above. The arrival of a large food parcel, whether it is a concentrated delivery of marine snow following a surface bloom, or a whale fall, or some other episodic input of organic material, triggers a response in the biological community that is detectable in subsequent observations. The abundance of amphipods, for example, may fluctuate significantly with the availability of food. During periods of food scarcity, populations may be lower and individuals more spread out. During and after periods of food abundance, populations can aggregate in large numbers around the food source, feeding intensively and then slowly dispersing as the resource is depleted. This dynamic behavior, the aggregation and dispersal of deep-sea organisms in response to the patchy and episodic supply of food, is one of the key adaptations that allow the Hadal ecosystem to function under chronic energy limitation. The organisms of the trench have evolved life history strategies and behavioral repertoires that allow them to take maximum advantage of food when it is available and to minimize energy expenditure when it is not. This ecological flexibility is as important as the biochemical adaptations to pressure and temperature. Without the ability to find and exploit food resources efficiently, the biochemical machinery for surviving at depth would be of little practical value. Among the many mysteries of the Mariana Trench, one of the most intriguing concerns the mechanisms by which organisms colonize and maintain populations in such an extreme and isolated environment. The trench is deep, narrow, and separated from other Hadal environments, other deep ocean trenches, by thousands of kilometers of abyssal seafloor at much shallower depth. How do organisms get there? How do they maintain genetic diversity in a population that appears to be physically isolated from other populations of the same species? And how do species distribute themselves within the trench? Are the same species found throughout its 2,500-kilometer length, or are there distinct communities at different locations? These questions of biogeography, the study of how organisms are distributed in space, are particularly fascinating in the Hadal context. On land, the distribution of species is determined by a combination of the species' ability to disperse, the availability of suitable habitat, the presence of physical barriers like mountains or rivers, and the historical patterns of past distributions and extinctions. In the deep ocean, these same factors operate, but in a very different physical context. Deep-sea organisms can potentially disperse through the water column by drifting as larvae or eggs in the water or by active swimming. But the deep trenches are in the horizontal dimension, isolated from each other by vast stretches of much shallower abyssal seafloor. For an organism specialized for Hadal depths, for the crushing pressure and specific conditions of the deep trench, The journey across thousands of kilometers of shallower seafloor would require passing through conditions that might be physiologically challenging or impossible. The pressure at abyssal depths, 3 to 6 kilometers, is very different from the pressure at Hadal depths, 6 to 11 kilometers. An organism adapted to the extreme pressures of the Mariana Trench would face a significant physiological challenge if it tried to migrate to another Hadal environment across the shallower seafloor of the abyssal Pacific. Yet molecular genetic studies of Hadal organisms have revealed something surprising. Many of the species found in the Mariana Trench are also found in other deep ocean trenches in the Pacific and elsewhere, and genetic analysis suggests that these geographically separated populations are not entirely isolated from each other. There is genetic exchange between populations in different trenches, evidence of occasional long-distance dispersal that connects populations across the barriers of shallower seafloor that separate the trenches. How this dispersal occurs is not entirely clear. One possibility is that the dispersal happens through the overlying water column, larvae or small individuals drifting up into shallower water, crossing the abyssal seafloor at depth ranges they can tolerate, and then descending into a new trench. Another possibility is that the dispersal happens through vectors that we have not yet identified, perhaps through association with mobile larger animals that move between Hadal environments or through some other mechanism. The existence of genetic connectivity between geographically separated Hadal populations is one of the more puzzling findings of recent deep-sea biology, and understanding the mechanism of this connectivity is an active area of research. Within the Mariana Trench itself, the question of how species are distributed along the length of the trench is also not fully resolved. The trench is not uniform along its length, conditions vary, terrain varies, and the supply of organic material from the overlying surface ocean likely varies with location. Do different parts of the trench host different communities? Are there species that are restricted to particular sections of the trench? Are the communities at the very deepest points, at the Challenger Deep, different from those found at slightly shallower locations within the trench? The answer, based on the available data, is that there is both commonality and difference across the length of the trench. Many species are found throughout much or all of the trench. The giant amphipods, for example, appear to be present at multiple locations along the trench length. But there are also differences in community composition between locations, and the deepest parts of the trench appear to have somewhat distinct communities compared to shallower locations within the Hadal zone. The extreme conditions at the very bottom of the Challenger Deep, the greatest pressure, the deepest isolation from the surface, the most severe of an already severe environment, appear to impose an additional filter on which organisms can survive there, producing a community that is a subset of the broader Hadal community with particular adaptations to the very deepest conditions. These questions of within-trench biogeography are important not just for their own sake, but for what they tell us about the ecological dynamics of the trench, about how populations are maintained, how the community recovers from disturbances, and how the ecosystem would respond to changes in the overlying surface ocean that might alter the supply of organic material to different parts of the trench. Understanding these dynamics requires the kind of systematic, repeat visit survey across multiple locations within the trench that the Fendouzhe program has been conducting, and that ongoing and future expeditions will need to continue. The contribution of the Mariana Trench and the broader deep ocean to the regulation of global climate is a topic that has received increasing scientific attention as the urgency of understanding climate processes has grown. The connection between the deep ocean and the climate system operates through several pathways, each of which is important in its own right and which interact with each other in complex ways. The most direct connection is through the carbon cycle. The ocean is the largest active reservoir of carbon on the planet's surface, containing roughly 50 times more dissolved inorganic carbon than the atmosphere, and an enormous additional quantity of organic carbon in the tissues of marine organisms and in the sediment. The exchange of carbon between the ocean and the atmosphere, particularly the exchange of carbon dioxide, is a major determinant of atmospheric carbon dioxide concentrations and therefore of climate. The ocean currently absorbs a significant fraction of the carbon dioxide emitted by human activities, slowing the rate at which atmospheric concentrations increase. Understanding the mechanisms and limitations of this ocean carbon uptake is critically important for projecting how the climate will respond to continued carbon dioxide emissions. The deep ocean plays a specific and important role in this carbon uptake through what oceanographers call the biological pump. The biological pump refers to the process by which carbon dioxide is fixed into organic matter by photosynthesis in the surface ocean, and then transported to depth through the sinking of organic particles and through the downward movement of dissolved organic matter and organisms. When this carbon reaches the deep ocean, it is effectively removed from contact with the atmosphere, sequestered in the deep for timescales ranging from decades to millennia, depending on how deep it goes and how quickly it is remineralized back to carbon dioxide. The efficiency of the biological pump, how much of the carbon fixed at the surface ultimately makes it to depth and how long it stays there, is a key parameter in determining the ocean's capacity to absorb carbon dioxide from the atmosphere. This efficiency depends on many factors: how productive the surface ocean is, what types of organisms are doing the photosynthesis, how much of the organic matter is consumed by organisms in the surface layer before it can sink, how quickly particles sink once they leave the surface layer, and how much remineralization occurs during the descent. The role of the Hadal zone, the Mariana Trench, and other deep trenches in the biological pump is a question that researchers are actively investigating. The Hadal zone is where the biological pump effectively ends, where the organic material that has survived the long journey from the surface finally reaches the seafloor. Understanding what happens to this material once it arrives, quantifying how much carbon is ultimately stored in the deep sea sediment for long timescales, and how much is respired back to dissolved carbon dioxide by the benthic microbial community. The finding that the microbial communities in Hadal sediments are more active than previously expected, that they are effectively processing and remineralizing organic carbon at rates that are significant relative to the input, has implications for our understanding of how the biological pump operates. If more carbon is being respired back to carbon dioxide at Hadal depths than our models assumed, then the net carbon sequestration achieved by the biological pump may be somewhat lower than previous estimates. Conversely, if some fraction of the organic carbon reaching the Hadal sediment is buried and preserved in the sediment for very long timescales, incorporated into the geological record rather than returned to the water column, then the Hadal zone may be an important long-term carbon sink. The balance between respiration and burial in Hadal sediments is not yet fully characterized. The data collected by the Fendouzhe and other programs provide some constraints, but a complete accounting requires more extensive sampling across the length of the trench and deeper investigation of the sediment column, drilling into the sediment to understand not just what is happening at the surface, but how organic carbon is preserved or transformed over the thousands of years of accumulation represented by the deeper sediment layers. The connection between the deep ocean and climate also operates through the thermohaline circulation, the global system of deep ocean currents driven by density differences in the water. This circulation, which includes the sinking of cold, dense water at high latitudes and the slow spreading of this water through the deep basins of the world's oceans, is responsible for the transport of heat and nutrients around the globe. It influences the climate of surface regions far from where the deep water originates by redistributing heat and by bringing nutrient-rich water back to the surface in upwelling zones that support some of the most productive fisheries on Earth. The deep water that fills the Mariana Trench is ancient. It has been out of contact with the atmosphere for a very long time, potentially thousands of years. This ancient deep water carries a chemical signature of the conditions that prevailed at the ocean surface when it was last in contact with the atmosphere: the temperature, the carbon dioxide concentration, the oxygen content. Measuring the chemical composition of the deep trench water provides, in effect, a sample of the ocean's ancient past, a record of what the deep ocean was like when the water now sitting at the bottom of the Challenger Deep was last at the surface. This retrospective view is one of the tools that oceanographers and paleoclimatologists use to reconstruct the history of ocean conditions, and through that, the history of climate. The sediment itself is also a record. The layers of sediment accumulated on the floor of the Mariana Trench over millions of years contain a detailed archive of past ocean conditions, preserved in the chemical composition of the sediment, in the fossils of organisms that lived in the overlying water column, and in the physical properties of the sediment layers themselves. Reading this archive is a major project of deep-sea geology and paleoceanography, and the sediment cores collected by the Fendouzhe and other missions are contributing valuable material to this effort. The question of what we will discover next in the Mariana Trench is, in some ways, impossible to answer. That is the nature of scientific exploration. If we knew what we were going to find, it would not be discovery. What we can say is that the history of deep-sea exploration has consistently delivered the unexpected, and that every increase in the technological capability to observe and sample the deep ocean has resulted in surprises that required revision of our understanding. The next generation of deep-sea exploration technology is already being developed, and some of it is beginning to be deployed. Autonomous underwater vehicles, robotic vehicles that can navigate without a cable connection to the surface, are becoming increasingly capable at deep ocean depths. Unlike remotely operated vehicles, which are connected to a surface ship by a cable that transmits power and communications, autonomous vehicles carry their own power supply and navigation systems and can operate independently for hours or days at a time. This independence from a tether greatly increases the range and flexibility of these vehicles, allowing them to cover much larger areas of the seafloor than a tethered vehicle can. Full ocean depth autonomous vehicles capable of operating in the Mariana Trench and other Hadal environments are being developed and tested. These vehicles, if they can be made reliable at Hadal depths, could transform the pace of deep-sea exploration by allowing multiple vehicles to operate simultaneously over large areas, building up comprehensive maps and observations at a rate that would be impossible with crude submersibles or tethered vehicles alone. The challenge is developing autonomous navigation systems that can function reliably in the deep ocean, where GPS signals do not penetrate and where the sonar-based navigation alternatives require careful engineering to be accurate enough for scientific purposes. Improved sensor technologies are also advancing the capabilities of deep-sea research. Mass spectrometers capable of operating underwater and at depth can identify chemical compounds in the water in real time, without the need to collect samples and bring them to the surface for analysis. This kind of in situ chemical sensing could dramatically improve our understanding of the chemical environment of the Mariana Trench, the distributions of nutrients, dissolved gases, organic compounds, and other chemical signals in the water above the trench floor and in the water within the sediment. Similarly, optical sensors that can detect and characterize bioluminescence, fluorescence, and other optical signals in the water could provide continuous maps of biological activity in the deep ocean without the need to physically collect organisms. Genomic analysis technologies are advancing at a particularly rapid pace. The ability to extract and sequence genetic material from environmental samples, water samples, sediment samples, has revolutionized microbial ecology, allowing researchers to characterize the diversity and abundance of microbial communities in any environment without the need to grow the organisms in culture. This technique, known as environmental genomics or metagenomics, is being applied increasingly to deep-sea samples and is revealing a level of microbial diversity in the deep ocean that is far greater than anything we suspected even a few decades ago. Future advances in this technology, faster sequencing, lower costs, the ability to analyze samples in situ rather than bringing them to the surface, will further accelerate the characterization of the microbial world of the deep sea. The development of instruments that can maintain samples at in situ pressure and temperature during recovery, bringing deep-sea organisms and sediments to the surface without exposing them to the dramatic changes in pressure and temperature that currently damage or kill many samples, is another critical technological frontier. If organisms can be recovered alive and intact from the Challenger deep, the possibilities for biological research are enormous. Culture experiments at simulated deep-sea pressure, biochemical studies of living deep-sea organisms, behavioral observations in pressure tanks, all of these possibilities would open up if reliable pressure-maintaining sample recovery technology could be developed. The combination of these technological advances, improved autonomous vehicles, better sensors, advanced genomic tools, pressure-maintaining sample recovery, will transform what is possible in deep-sea research over the coming decades. The Mariana Trench, which has been explored in only the most superficial sense by the standards of what future technology will make possible, will yield its secrets with increasing speed. Not quickly by our everyday standards, this will still be the work of decades and generations, but at a pace that will dramatically accelerate the accumulation of scientific knowledge about the deep ocean. There is a temptation, when describing the extraordinary discoveries made in the Mariana Trench and the deep ocean more broadly, to frame these discoveries as existing purely in the realm of the exotic and the remote, as amazing facts about an alien world that has no direct connection to our everyday lives. This framing is understandable but ultimately misleading, and it is worth taking some time to correct it. The deep ocean is not separate from us. The processes occurring in the Mariana Trench, the cycling of carbon, the processing of organic material, the harboring of biodiversity in extreme conditions, the function of the deep ocean as a repository of chemical and biological information, these are not abstract scientific concerns. They are parts of the planetary life support system on which all life on Earth, including human life, depends. Consider the carbon cycle. We live in a period when the concentration of carbon dioxide in the atmosphere is rising, as a result of the combustion of fossil fuels and other human activities. This rise in carbon dioxide is driving climate change, warming temperatures, shifting precipitation patterns, rising sea levels, and changes in the frequency and intensity of extreme weather events. The ocean absorbs a substantial fraction of the carbon dioxide we emit, slowing the pace of atmospheric accumulation. The deep ocean, including the Hadal zones of the world's great trenches, plays a role in this carbon absorption. If the capacity of the deep ocean to absorb and store carbon changes, as it might as ocean temperatures rise and circulation patterns shift, this will affect the trajectory of climate change. Understanding the mechanisms of deep ocean carbon cycling, including the role of the Hadal zone, is therefore directly relevant to understanding the future of the climate and the pace and severity of climate change. Consider biodiversity. The organisms living in the Mariana Trench are the products of millions of years of evolution under some of the most extreme conditions on Earth. The biochemical adaptations they have developed for surviving under crushing pressure, for maintaining biological function in the cold and dark, for extracting energy from chemical rather than photosynthetic sources, represent solutions to engineering problems that human science has not yet solved. The TMAO-based pressure stabilization system used by deep-sea fish, the membrane adaptation strategies of Hadal bacteria, the enzymatic innovations that allow deep-sea organisms to maintain biological function at temperatures and pressures that would stop most biochemistry cold. These are nature's solutions to problems that we are just beginning to understand. Some of these solutions may have practical applications. The enzymes used by microorganisms living in extreme environments, what biotechnologists call extremozymes, have already proved enormously valuable in a variety of industrial and medical applications. The DNA polymerase enzyme used in the polymerase chain reaction, the technique that forms the basis of most modern molecular biology and genetic testing, was originally derived from a bacterium living in hot springs. Similar enzymes from deep-sea pressure-adapted organisms might have valuable properties for applications requiring biochemical activity under conditions that would destroy conventional enzymes. The study of deep-sea biochemistry is not just basic science, it is a potential source of technological innovation. The biodiversity of the deep ocean also represents a reservoir of genetic information that is irreplaceable. The organisms of the Mariana Trench carry in their genomes the record of millions of years of evolution under conditions unlike any other on Earth. This genetic information, encoded in the sequences of their DNA, contains potential insights into the mechanisms of life itself, into how biological systems are built, and how they can be modified to function in different environments. Once lost, this genetic information cannot be recovered. The extinction of a deep-sea species, by whatever cause, whether by the degradation of its habitat through pollution or ocean acidification, or the disruption of the surface ocean productivity that supports the deep-sea food chain, represents the permanent loss of a unique and potentially invaluable biological heritage. This concern about the vulnerability of the deep-sea ecosystem is not hypothetical. The discovery of microplastics in the sediment of the Challenger Deep demonstrates that human pollutants are reaching even the most remote environments on Earth. Ocean acidification, the decrease in ocean pH caused by the absorption of carbon dioxide, affects the chemistry of the deep ocean as well as the surface, and may have consequences for organisms that build carbonate shells or skeletons. The effects of climate change on ocean circulation could alter the supply of organic material to the deep sea. The potential future development of deep-sea mining, the extraction of mineral resources from the deep-sea floor, raises the possibility of direct physical disturbance of deep-sea habitats that have been essentially undisturbed for geological ages. The deep ocean has generally been outside the scope of conservation discussions that focus on more visible and familiar environments, tropical forests, coral reefs, iconic land animals. But the deep ocean is as much a part of the planet's biosphere as any surface habitat, and it deserves the same attention and the same protection. Understanding what is down there, mapping its biodiversity, characterizing its ecological function, measuring its contribution to planetary processes, is the necessary first step toward any meaningful effort to protect it. The philosophical implications of what has been discovered in the Mariana Trench extend beyond the purely scientific. They touch on questions about the nature of life, the nature of the planet, and our place within the larger system of the biosphere. For most of human history, the image of life on Earth was essentially a surface image. Life was what we could see: the forests and the fields, the animals and the insects, the bacteria that made us sick and the fungi that decomposed our food. The ocean was a surface phenomenon, a vast expanse of water on which ships sailed and from which fish were taken, with a depth that was largely irrelevant to human experience and understanding. The idea that the vast majority of the habitable volume of Earth, the deep ocean, was largely empty and biologically insignificant was comfortable, in a way, because it meant the world was more or less the size and shape we imagined it to be. The exploration of the deep ocean has shattered this comfortable image. It has revealed that the world is enormously larger in biological terms than we thought, that life pervades environments we considered uninhabitable. That the deep, dark, pressurized world beneath the ocean's surface is as richly alive as any surface habitat, adapted in ways we are only beginning to understand, connected to the surface world through processes we are only beginning to trace. This revelation carries with it a kind of humility, a recognition that our knowledge of our own planet, the planet we have lived on for our entire existence as a species, is far more limited than we like to assume. We have mapped the surface of the moon in greater detail than we have mapped the floor of the Mariana Trench. We have sent spacecraft to the outer planets and returned images of their moons and rings and atmospheric storms. We have peered through telescopes at galaxies billions of light years away. And yet the deepest point on our own planet remained essentially unvisited for decades between the first crude descent in 1960 and the resumption of systematic exploration in more recent years. There is something worth sitting within this fact, not as a criticism. The engineering challenges of reaching the bottom of the Mariana Trench are genuinely formidable, and the pace of deep-sea exploration has been constrained by real technological and logistical limitations, but as a reflection on the relationship between accessibility and knowledge. We tend to explore what is most accessible, most visible, most amenable to the technologies we already have. The deep ocean has been difficult to explore, and so it has remained largely unexplored. The difficulty of the exploration has translated directly into a gap in our knowledge of the world we live in. Closing this gap, building the technology to explore the deep ocean systematically and comprehensively, and deploying that technology in a sustained and well-funded program of research is one of the most important scientific projects of our time. Not because the deep ocean is more important than other frontiers of science, but because it is a frontier of our own planet, a frontier on which our own well-being depends in ways we are only beginning to appreciate. The organisms living at the bottom of the Mariana Trench do not know that they are being studied. The amphipods swarming the sediment, the snailfish gliding through the dark water, the archaea processing organic carbon in the sediment, none of them are aware of the scientific expedition descending toward them from the surface. They are simply living their lives as they and their ancestors have been living them for millions of years, in the dark and the pressure and the cold, engaged in the ancient, fundamental project of being alive. It is we who are changed by the encounter. It is we who leave the experience of deep-sea exploration with new questions, new perspectives, and new appreciation for the extraordinary scope and depth of the living world. The Mariana Trench does not need us to understand it, but we need to understand it for the sake of science, for the sake of the planet, and perhaps most importantly, for the sake of the particular kind of wisdom that comes from confronting the limits of our own knowledge. As the program of Fendouzhe dives has continued, and as the body of data and observations from the Mariana Trench has grown, researchers have begun to work towards something that had not previously been possible: a coherent, quantitative model of how the Challenger Deep and the broader Mariana Trench ecosystem function as integrated systems. This modeling effort, which draws on the biological observations, the chemical measurements, the physical oceanographic data, and the geological information gathered during multiple expeditions, is still in its early stages, but the outlines of a comprehensive understanding are beginning to emerge. The picture that is emerging is one of a deeply interconnected system. The deep trench ecosystem is connected upward to the surface ocean through the biological pump, the sinking of organic material from the photic zone. It is connected horizontally to the surrounding abyssal seafloor through the movement of organisms and the transport of chemical signals in the bottom water. It is connected downward to the geological substrate through the chemical exchange between the water and the sediment, and through the tectonic processes that created the trench and continue to shape its geometry. And it is connected, through the carbon cycle and the thermohaline circulation, to the climate system and to the global ocean as a whole. Within this connected system, the trench plays multiple roles simultaneously. It is a biological community, a complex ecosystem with its own food web, its own diversity of organisms, its own ecological dynamics. It is a biogeochemical processor, a site where organic carbon and other biologically important elements are transformed and cycled through a series of reactions. It is a geological archive, a repository of sediment that records the history of ocean conditions and climate over millions of years. And it is an extreme environment, a place that has pushed the evolution of life to the limit of what is biochemically possible and has produced in the process some of the most remarkable adaptations in the history of the biosphere. Understanding all of these roles and understanding how they interact with each other and with the broader planetary system is the goal of the ongoing and future research program. It is a goal that will not be achieved quickly. The Mariana Trench is complex, the data collection is difficult, and the integration of data from multiple disciplines into a coherent model is a scientific challenge of the first order. But the progress that has been made, the progress documented in this film, from the initial discovery of abundant life at extreme depths to the detailed characterization of molecular adaptations to the revelation of the ecosystem's connection to planetary systems, represents an extraordinary scientific achievement. The scientists who have made this progress have done so with great dedication and creativity, working at the frontier of what technology permits and pushing that frontier forward through their own ingenuity and determination. They have built submersibles capable of surviving at the deepest point on Earth and have ridden in them to the bottom of the world. They have designed instruments capable of measuring the chemistry of water and sediment at extreme depth. They have developed analytical techniques for extracting biological information from vanishingly small samples of deep-sea material. They have spent careers studying an environment that most people have never seen or thought about, driven by the conviction that understanding the deep ocean is important for science, important for the planet, important for our collective understanding of what it means to live on a water world. As we approach the conclusion of this journey through the Mariana Trench, this two-part exploration of the deepest place on Earth and the extraordinary life it harbors, it is worth pausing to look back at how far our understanding has come and forward at how far it still has to go. We began with the assumption that the deep ocean was empty. We began with the image of the Mariana Trench as a lifeless void, a geological feature of extreme depth and extreme conditions that represented the ultimate limit of what the Earth's environment could impose and therefore the ultimate limit of where life could exist. This assumption was wrong, but it was not unreasonable. It reflected the best available scientific understanding of the early 20th century, and it was consistent with everything that was known about life and its requirements at the time. What has overturned this assumption is not a single dramatic discovery, but a long series of expeditions, observations, experiments, and analyses that have gradually built up an entirely different picture. The bathyscaphe Trieste provided the first crack in the assumption of a lifeless deep. The discovery of hydrothermal vent ecosystems in 1977 proved that life could exist in the complete absence of sunlight, given a suitable chemical energy source. The growing body of observations from deep-sea submersibles and landers revealed organisms living at depths and pressures far beyond what early predictions allowed. The molecular biological analysis of deep-sea organisms revealed the extraordinary biochemical mechanisms that make this survival possible. And the most recent expeditions, the Fendouzhe program and others, have provided the most comprehensive and detailed picture yet of what actually lives at the bottom of the world and how the deep-sea ecosystem functions. The picture that has emerged from all of this is one of remarkable biological richness in an extreme environment. It is a picture of life not as a fragile, easily disrupted phenomenon that clings to existence only in the narrow range of comfortable conditions we are familiar with, but as a robust, adaptable, extraordinarily creative process that has colonized every accessible environment on Earth. It is a picture of the biosphere as something far larger and more complex than our surface-dwelling intuitions suggest, a biosphere that extends to the very bottom of the ocean's deepest trenches, and that maintains its fundamental character, eating, growing, reproducing, evolving under conditions of pressure and darkness that would seem, from a surface perspective, to make all of this impossible. The lesson of the Mariana Trench is, in a profound sense, a lesson about the nature of life itself. Life is not a thing that simply occupies environments, it is a process that transforms environments. It takes inorganic chemicals and turns them into biological structures. It takes solar energy or chemical energy and converts it into the organized complexity of living systems. It generates, through the mechanism of evolution, solutions to environmental challenges that could not have been predicted in advance and that continue to surprise us when we find them. Life is not fragile. It is creative, it is adaptive, it is persistent beyond anything we might have imagined before we began to look carefully at the places where it is pushed to its limits. The Mariana Trench does not prove that life can exist anywhere. There are still environments where no life has been found, in the cores of asteroids, in the upper atmosphere, in the hottest portions of hydrothermal vents. But it pushes the apparent limits of life so far beyond what we once thought possible that any future claim about where life cannot exist must be made with extraordinary caution. We have been wrong before. We have assumed that life cannot survive at crushing pressure, in total darkness, under conditions of extreme cold and chemical strangeness, and we have been spectacularly wrong. Any future claim about the limits of life must reckon with the lesson of the Mariana Trench. There is one more dimension of the Mariana Trench story that deserves attention before we conclude, and it is perhaps the most quietly profound dimension of all, the dimension of time. The Mariana Trench is old. It has existed in approximately its current form for tens of millions of years, far longer than our species has existed, far longer than any of the great civilizations of human history, far longer, even, than the ancestors of modern humans had fully separated from the lineages leading to our nearest primate relatives. For all of that time, for tens of millions of years, the trench has been sitting in the western Pacific, collecting life, accumulating sediment, cycling nutrients, responding to the slow changes in the overlying surface ocean while maintaining the fundamental character of an extreme, dark, pressurized, deep environment. The organisms living at the bottom of the Mariana Trench have been there, in one form or another, for most or all of that time. Not the exact same individuals, individual organisms are short-lived even in the deep sea. Not even exactly the same species evolution has been operating continuously, modifying populations over time, but organisms of similar lineages, adapted to similar conditions, living similar lives in this same place for geological ages. When the Fendouzhe descends and its lights sweep across the sediment of the Challenger deep, it is illuminating a world that has been dark and alive for longer than the human mind can truly comprehend. This temporal perspective, this sense of the deep time of the trench and the communities that live in it, is one of the most important gifts that deep-sea exploration can offer. It connects us, in an immediate and visceral way, to the geological history of the planet. It reminds us that the world we live in is not a static backdrop to human activity, but a dynamic, evolving system that has been changing continuously for billions of years, and that will continue to change long after we are gone. It reminds us that the particular moment in the planet's history in which we find ourselves, the moment of human civilization, of industrialization, of global environmental change, is a brief episode in a much longer story. And it suggests that the consequences of what we do in this brief episode may ripple forward through time in ways that affect the very deep places we have only just discovered. The microplastics in the sediment of the Challenger deep are, in this temporal frame, a new addition. They arrived recently, within the last several decades, the geological equivalent of an eye blink, but in the sediment record they will persist. Future geologists, human or otherwise, who drill into the sediment of the Challenger deep millions of years from now, will find a layer of synthetic polymer particles that marks the beginning of the period when one species on Earth's surface began to produce materials that had no precedent in the planet's chemistry and that distributed themselves to every corner of the biosphere. This layer will be as clear a marker in the sediment record as the iridium-rich layer that marks the impact of the asteroid that ended the age of the dinosaurs. This is the scale on which the deep ocean thinks, if we can speak metaphorically of a geological feature as thinking. The deep ocean operates on timescales of millions of years. It will absorb what we do to it and continue, changed in ways we may not be able to predict, but continued. The question is what the community of organisms at the bottom of the Mariana Trench will look like after absorbing those changes. Whether the extraordinary, ancient, biochemically brilliant organisms that have survived for geological ages at the bottom of the world will find a way to adapt to the novel challenges we are introducing. Whether the deep-sea food web will be disrupted by the accumulation of pollutants or the reduction of organic material supply from a changing surface ocean, or whether it will prove as resilient as it has been to the other changes the planet has thrown at it over tens of millions of years. We do not know the answers to these questions. We are adding stressors to a system we do not yet fully understand, and the outcomes of that experiment, the largest and most consequential scientific experiment in human history, conducted not in a laboratory but on the actual planet, are not yet clear. What is clear is that we have a responsibility to understand what we are doing, to monitor what is happening in the deep ocean, to characterize the baseline conditions of the ecosystems there before they are further changed by our activities, and to use that understanding to make better decisions about how we interact with the planetary system that sustains us. The Fendouzhe program and the broader community of deep-sea science it represents is part of this effort. Every dive into the Mariana Trench, every sample collected, every measurement taken, every organism documented adds to the baseline of knowledge that we will need to understand the state of the deep ocean going forward. Every advance in the technology of deep-sea observation extends our reach and our capability. Every paper published, every dataset shared, every image released of the extraordinary world at the bottom of the ocean expands the circle of people who know what is down there and why it matters. This is how science works, not in single dramatic revelations, but in the slow accumulation of knowledge, dive by dive, sample by sample, measurement by measurement, insight by insight. It is slow and it is incomplete, and it requires patience and dedication and willingness to work for years toward goals that can never be fully achieved. But it is the only way we have to understand a world as complex and as important as the deep ocean. And the progress that has been made from the first sounding by HMS Challenger in 1875 to the systematic exploration being conducted by the Fendouzhe today is genuine, substantial, and profoundly important. Let us take one final descent, not in a submersible this time, but in the mind. Let us imagine the Challenger deep as it exists right now in this moment while you are watching this film. While you are sitting in your home or your office or wherever you happen to be, the floor of the Mariana Trench is going about its business in the dark. The amphipods are there, moving across the sediment in their hundreds and thousands, probing the seafloor for organic material, occasionally pausing to feed on a fragment of marine snow that has drifted down from above. Their bodies, adapted over millions of years, handle the 1,100 atmospheres of pressure with the ease of an organism that has never known any other condition. For them, this pressure is normal. It is the pressure of home. The snailfish are there, gliding through the water column just above the sediment, their pale, translucent bodies moving through the darkness with a slow efficiency that reflects the energy economy of an environment where food is hard to find and nothing can be wasted. They are hunting amphipods, feeling the pressure waves through their lateral line system, zeroing in on the movements of their prey with a precision that millions of years of evolution in the dark have refined to a high art. Their muscles and enzymes and membranes, all carefully tuned to function at the greatest depth any fish has ever lived, operate flawlessly. The bacteria in the sediment are there, countless billions of them in every gram of sediment, cycling carbon and nitrogen and sulfur in reactions that connect the biology of the trench to the chemistry of the deep ocean, and through that to the chemistry of the atmosphere and the climate of the planet. They work without awareness, without intention, without any sense of the importance of what they are doing. But what they are doing matters enormously. It is part of the machinery that regulates the chemistry of the planetary surface and keeps it habitable for life. The sediment itself is there, layer upon layer of accumulated particles stretching back through time, recording in its chemistry and its fossil content the history of the ocean and the climate over millions of years. Somewhere in those layers, a stratum of microplastic particles marks the arrival of the industrial age. The sediment does not judge this intrusion. It simply records it as it has recorded everything else that has settled to the bottom of the world. And above all of this, 11 kilometers of dark water stretch upward toward the surface, toward the light, toward the world where we live. The surface ocean is up there, turbulent and sunlit and alive with its own forms of life, connected to the deep trench below by the slow rain of organic particles and the periodic dramatic deliveries of whale falls and other concentrated nutrients. The two worlds, the surface world of sunlight and weather and familiar biology, and the deep world of pressure and darkness and extraordinary adaptation, are not separate. They are the same world seen from different depths. We are part of this world. We are part of the planetary system that includes the Mariana Trench and the Challenger Deep, and the organisms living at the bottom of the world. Our activities affect that system, and the system affects us. The deep ocean absorbs our carbon dioxide and moderates our climate. The organisms living at the bottom of the world cycle nutrients that eventually return to the surface and support the productivity that ultimately feeds us. The genetic information encoded in the genomes of deep-sea organisms contains biological solutions that we may someday need. We are connected to the bottom of the world whether we know it or not. What has changed, what the exploration of the Mariana Trench has given us, is the knowledge that the bottom of the world is not empty, the knowledge that life extends to the very greatest depths the planet offers. The knowledge that the extraordinary adaptations of deep-sea organisms demonstrate the creative power of evolution applied to the most extreme conditions imaginable. The knowledge that the deep ocean is connected to the surface world in ways that matter for the climate and the carbon cycle and the health of the planetary system. Knowledge is not sufficient by itself. Knowledge requires application, requires the wisdom to understand what the knowledge means and the will to act on that understanding. But knowledge is where it begins, and the exploration of the Mariana Trench, the long, difficult, expensive, extraordinarily rewarding project of understanding the deepest place on Earth, is in the most fundamental sense an act of knowledge-seeking, an act of curiosity pursued to its most extreme conclusion. The trench is there, it has always been there, and it has always been alive in the dark, patient and ancient and extraordinarily complex. We are only now beginning to know it. And knowing it, truly knowing it, in all its depth and complexity and connection to the rest of the world, will take generations of scientists and engineers and explorers who have not yet been born. But the work has begun. The window into the abyss has been opened. The lights of the Fendouzhe have swept across the sediment of the Challenger Deep and revealed, in that narrow cone of illumination at the bottom of the world, that we are not alone on this planet. That life is here with us in every corner and every extreme and every depth the Earth has to offer. That the world we live on is so much more alive, so much more complex, so much more full of wonder than we ever imagined. The bottom of the world is not empty, it never was. And the more we look, the more we discover just how extraordinary, how ancient, how connected, and how vital is the life that has been living there in the dark, all along, waiting for us to find it. This has been the story of the Mariana Trench. The story of the deepest place on Earth, the extraordinary life it harbors, and the ongoing human effort to understand it. It is a story that is still being written, one dive at a time, at the bottom of the world. And if the history of deep-sea exploration has taught us anything, it is that the next chapter, the next dive, the next discovery, the next revelation from the darkness will be something we did not expect. The deep is patient. The deep is ancient. The deep is alive. and we have only just begun to listen to what it has to tell us. There is a place on this earth so deep, so dark, and so utterly removed from the world we know, that for most of human history we simply assumed nothing could live there. We assumed the pressure would crush every living thing. We assumed the cold would still every biological process. We assumed the darkness would extinguish every spark of life before it ever had a chance to ignite. We assumed, in short, that this place was dead. That it was nothing more than an enormous, silent grave carved into the bottom of the ocean floor, a wound in the planet so severe and so ancient that even nature itself had given up on filling it with anything meaningful. We were wrong. We were profoundly, spectacularly, and almost embarrassingly wrong. What lies at the bottom of the Mariana Trench, at the very deepest point on the surface of this entire planet, is not emptiness. It is not silence in the way we once imagined silence. It is not a void. What lies down there, waiting in the crushing dark beneath nearly 11 kilometers of seawater, is life. Extraordinary, resilient, bewildering, almost unbelievable life. Life that has found ways to not merely survive, but to thrive in conditions that would destroy any organism we might consider familiar. Life that has rewritten the rule book on what biology is capable of. Life that has been quietly going about its business for millions of years while we, on the surface, looked down into the ocean and assumed there was nothing worth looking for. This is the story of the Mariana Trench. This is the story of the Challenger Deep, the single deepest point ever measured on the surface of this earth, a location so extreme it sits nearly 7 miles beneath the surface of the Pacific Ocean. This is the story of the machines built to reach it, the scientists brave enough to descend toward it, and the living world they discovered waiting for them when they arrived. This is the story of what happens when human curiosity refuses to accept a boundary, even one drawn at the very bottom of the world. And this is, perhaps most importantly, the story of how much we still do not know. Because for all the progress that has been made, for all the remarkable descents and the extraordinary footage and the astonishing biological specimens hauled up from those crushing depths, the truth is that the Mariana Trench mechanisms by which organisms colonize and maintain populations in such an extreme and isolated environment. The trench is deep, narrow, and separated from other Hadal environments, other deep ocean trenches, by thousands of kilometers of abyssal seafloor at much shallower depth. How do organisms get there? How do they maintain genetic diversity in a population that appears to be physically isolated from other populations of the same species? And how do species distribute themselves within the trench? Are the same species found throughout its 2,500-kilometer length, or are there distinct communities at different locations? These questions of biogeography, the study of how organisms are distributed in space, are particularly fascinating in the Hadal context. On land, the distribution of species is determined by a combination of the species' ability to disperse, the availability of suitable habitat, the presence of physical barriers like mountains or rivers, and the historical patterns of past distributions and extinctions. In the deep ocean, these same factors